Okay, all right. So, good evening. So, some of you might be joining up to speed sessions for the first time today, and some of you might be joining for the first time because you missed the particular section that we are covering today, and then you want to cover that section, and then you go about in your normal theory class life. So, either way, that is the way I want you to work. That is the way I want you to work. That I want you to get the benefit from what I am doing, which is this is an extra program. Okay, so I want you to use it as selfishly as possible. Selfish means, of course, you pay for my service, therefore you should be able to extract, or you should be able to meet your requirements in any way possible from me. Okay, so I started this up to speed sessions with the main intention of guiding and also sort of bringing my new students who are joining the theory class completely oriented into the A-level pattern, and also my teaching as well as the pattern you should expect in English medium A-level biology. So all those things we have been doing nicely for the past. Now this is our day 6 or 5, day 5, no? Yeah, day 5. And we have gone from the first lesson. Yes, we have already covered the first lesson. First lesson, by now you should know the first lesson is supposed to be only, if you take the teacher's guide, it's only 2 periods. Okay, so the first lesson is something we don't even, it's not a very considerable lesson. No? So there's going to be at least just one MCQ and a small part of a structured essay from the first lesson. Of course, we have covered it. And I have sent the playlist of all the up to speed sessions that we have done to groups. So if you just go to the first recording, you will be able to see our first lesson coverage done on the up to speed sessions. Okay, so yeah, so we are almost finishing cellular organelles, neither. So yeah, so this is what we have been doing the last week. So we started, we started right here with the plasma membrane, and then we moved on ahead with all the organelles, and we finished cytoskeleton. And today we have to move on from our remaining cellular organelles. And my plan today is that we are going to move on to the cell cycle, and we are going to, if we have, the plan today is to finish mitosis. Okay, because otherwise you have a small paper coming up for Thursday group people. It is going to be on 13th, and for Saturday people it's going to be on 15th. So before that session, before that paper, I want everyone to be in the same level. Therefore that's why I have to conclude up to speed sessions there onwards. You guys have the theory class which is going ahead as planned with the normal plan of covering our, as we go ahead. Okay, so there's going to be, in other news, there's going to be the starting of the new lesson, evolution and diversity, third lesson. That is going to happen for Saturday people this Saturday, and for Thursday people next Thursday. Okay, so there's going to be a new tute that will be delivered to you as soon as possible. I have already sent it to the post, so that it's in the hands of the postal department. It will be delivered to you as soon as possible. Okay, so yeah, so I'll be, yeah, you can listen in the tute. So this is only for online people, Harry. This is only for online people. I know some of my theory people are, physical people are also there. This is only for online people. In the next tute package that you will receive, Harry, in the next tute package you will receive, you will have the special exam paper but in a separate envelope. Harry, you will have the exam paper in a separate envelope inside the tute package. Okay, so I want you to not open it until the exam day, because one thing, we need to time it. Okay, so you should join the Zoom and then only you should start writing it. That is one thing. And the other thing, you should do it in an exam way. Even though you are at home, I want you to be honest and be, let's say, do it properly. That's it. That's all I'm expecting. Okay, so in order to do that, keep the envelope with the question paper separately. Hurry, keep it separately. Just take out the normal theory class tunes that will be there and join with the theory classes this week as usual. Then on 13th, you can face the exam once these up to speed sessions are over. Hurry. So that is all you need to know. Anyone brand new? Okay. So you should be, as soon as possible, if you are planning to join online, please go and enroll to the website Gurutaru Online. There you will be able to access recordings. Tube delivery will be done, and then the Zoom links will be available there. Physical people, of course, you are going to meet me now. So any issues, you can contact. And I have again and again sent my hotline numbers. They are always there for you to help with anything. Any issue that you have, tube delivery, class joining, recordings, payment, any other issue. Okay, if you want to directly ask me anything, they will help you with that. Any doubt that you have, they are always there to help you. So online, physical, whatever, you will not be, kept in the dark. You can always ask anything. You can always clarify your doubts, Hari. So okay, with that said, anything you want to ask, you can keep the Zoom chat active, and you can ask anything that you want clarified. And okay, so shall we move on? Anyone who doesn't have the tute that we are doing right now, if you are a brand new man or a woman, you can raise your hand. Anyone who doesn't have this? Okay, I'll send to the Zoom chat. Okay, and please get it downloaded, and it will be delivered to you together with. If you enroll this month, it will be delivered to you, of course. Okay, where is it? Audio, video, all clear, ne? I couldn't ask. You can hear me and see me well. Okay. Neither? You can. Okay, the diagram tute. So we have two types of tutes. Of course, you need to. So new people don't know. So we have two types of tutes that we complete in our class. Okay, so you will be getting a question tute that will have practicals as well. Then our diagram tute is the one with all the diagrams that you see on the board. Okay, so both will be, that is my actual question pattern for my 27 people also. They are following a similar structure. Okay, so they are following a very similar structure with a nice diagram tute with all the things that they need to understand the lesson properly, and to make the short note in. Okay, so that you don't have to waste time drawing diagrams. It will all be accessible through the, made easier with the tube. So this is something I did for my 27 people. There was a special immunity camp, which is a difficult lesson that they think. Okay, so we covered that last Sunday. Anyway, so for you guys also, such syllabus coverage programs are going to happen every now and then. Okay, so because biology is syllabus coverage before anything. Okay, but at the same time, you need to go with that syllabus coverage so that you have understood. Okay, the train has gone, you are not on the train. Syllabus has been covered, but you were not there when the syllabus got covered in usual theory classes. Okay, so that won't happen if you follow in the way I want you to follow, and if you complete the tutes, if you do the homework essays that I give you, then there is no such an issue. Hurry. Okay, anyway, everyone take out the resource book. Anyone who doesn't have the resource book? Anyone who doesn't have the resource book at this point? PDF or whatever, PDF. Anyone has, that's good. Okay, everyone has. Hurry, shall we come to, so we finished cytoskeleton. Hurry, you finished cytoskeleton. The playlist is available in the chats. I'll be setting it again as well. Okay, the playlist of all the up to speed sessions are available in the groups. Okay, also in the website they are available in your respective courses. So Thursday and Thursday course with Saturday and Saturday course it's available. Okay, hi. Then okay, we'll move on to cilia and flagella, which is our starting point. Okay. Shall we read? Come to the resource. Let's read what is there in the resource, page number 36, cilia and flagella. Cilia and flagella share a common structure. Okay. What do you mean by cilia and flagella? If you take cells, they are, so if you are a unicellular organism, that means as an organism you have only one cell. Okay, for example, if you take a mehema bacteria, salmonella typhi wage ohom ekkenek gatthoth, he is going to have this sort of a shape. Together with that, he is going to have some these sort of projections that are going to help him move. Those are his locomotive structures. Like you have legs, they are going to have this flagella as their locomotive structures. Like a boat has a motor on the outside, that is going to move the boat. Okay, so cilia, so mainly flagella. So if you in your body, do you have any cells that are flagellated? In your body, do you have any cell that is that are flagellated? Yes, anyone? Any cell that is flagellated in your body? Any cell that is flagellated? The sperms. Yeah, only males are able to produce this. There is only cell in your body that are flagellated, that are able to move. Alright. So sperms are designed to swim through the female reproductive tract and reach the egg. Okay. So sperms have a long flagellum. And if you take cilia, if you take the surface of the cell, cilia are a bit smaller. Cilia are very, compared to flagella, cilia are much smaller. Cilia are very, compared to flagella, they are very small, but they are more numerous. You have a lot of cilia. And the cilia are like they are very coordinated. Okay, they are like a dance team. You have seen on the swimming pool surface there are dancing again, rhythmic something, right? So your cilia are designed to move in a rhythmic way. Okay, so that they are able to move or sweep. They are rhythmically able to beat one side and the other. They go and go either way. So cilia can beat. That means wave like rhythmic motions. So when that happens, usually if there is a fluid, on the surface of the cell, certain fluid, there is a fluid on the surface of the cell, these cilia are able to move it. These cilia are able to move that fluid over the cell surface. Okay, so we'll see some of the examples. Okay, got it. So now what we are going to do is we are going to discuss the sort of the very microscopical structure of the cilia. I have given here electron microscope, that is actually centriole, which is having a very similar structure. Okay, now clear. Okay, let's read what is given in the resource. Cilia are long elongated and flagella are long elongated. Cilia are short cellular that are often organized in rows. Can you highlight? Flagella are long and elongated. Cilia are short that are often organized in rows. Then cilia are more numerous. You have more cilia. If you flagella wise, you have only maybe maximum 1, 2, 3 flagella. Okay, that much. But if you have cilia, numerous, hundreds of cilia. They are made up of microtubules. Now, what are microtubules? You might ask me. What are microtubules? What do you mean by a microtubule? Maybe we learned them last week. We learned about microtubules last week. Okay, these are microtubules. Metin, this is a microtubule. Okay, it's a tube. It's a microscopic pipe. It's a long tube. PVC, but right? So it is composed of a protein called tubulin, and it has a tube-like shape. Okay, so it is the cilia and flagella are composed of microtubules, which is this structure. Okay, so they are both made up of microtubules. Then listen carefully. With a 9+2 structure. What do you mean by 9+2? You can see here. Methi. This is our 9+2 structure. This is our 9+2 structure. Okay. What do you mean by 9 is, now listen carefully, something confusing a bit. Okay. there are 9 doublets. Doublets means what? Double microtubules. That means they are connected with each other. Double microtubules arranged in a ring. Okay. 9 doublets of microtubules are arranged in a ring. Okay. Then at the center you have two single microtubules. Again, they are not connected with each other like these ones on the around them. Okay, so you have 9 doublets in a ring, and at the center you have two central single microtubules. Okay, so two microtubules in the center. So we call this arrangement as 9 plus 2 because you have 9 on the surround in a ring and you have 2 in the center. Correct? So that's what we call as 9 plus 2 structure. So you can see if you zoom out, now this is the plasma membrane. This is the plasma membrane of the whatever big cell. Correct? Then coming out of the plasma membrane, you have our this is our flagellum. We have the flagellum. So we have cut it here so that we can see it. So usually the flagellum is very long. Okay, if you take the sperm cell, a sperm has a very long flagellum. Ne? So this has been cut in the center. So now you can see in a 3D view. Ne? You can see in a 3D view if you zoom in. You can see in this diagram, remember 9 doublets of microtubules are there with two grey ones. Can you see two grey microtubules at the center? That is what we call as 9 plus 2 structure. Cilia also, even though they are small, they have the same structure. Cilia also, even though they are small, they have the same structure. Hurry. They are covered by plasma membrane and bound to a basal body. Okay. Now I want you to, your resource book handling, I think I have talked about this. Keep it nicely and I want you to highlight with me nicely the resource book. Okay. So you are highlighting permanently in the keep it neat. Okay. Did you highlight? They are made up of microtubules with a 9 plus 2 structure. Highlight 9 plus 2, very important. Then they are covered by plasma membrane and bound to a basal body. Can you write covered by plasma membrane in the place it says covered by plasma membrane? If you don't like to write on the resource book with a pen, then you can write with a pencil. Put an arrow and write in eukaryotic cells. Covered by plasma membrane in eukaryotic cells. Eukaryotic means like your cells, plant cells, fungal cells, so animal cells. Those kind of cells you have, they are covered by plasma membrane. Okay, so you can see this flagellum has a plasma membrane covering. This flagellum has a plasma membrane covering. So that's why but it's only seen in eukaryotic cells, not in bacteria, those kind of cells. Prokaryotic cells you can't see. Then listen very carefully. Bound to a basal body which anchors the cilium or the flagellum. Highlight basal body. Basal body. So imagine now if you have a plug, right? If you have a electrical plug, then to plug in your TV or something, you have a plug like this with the three sort of the three pins. Then on the wall you have, so this is the plug. Okay, this is the plug. On the wall you have the plug base. This cilium or a flagellum should be connected or plugged into the cell in a proper way. So that is done by a special structure that you find on the, in the cell, that is where the flagellum or the cilium connects to the cell, that is called as, yes, Kiana, what do you call that? Basal body. It is like the root of a tree. Okay? This is the root of the cilium or the flagellum. Okay? So it is also composed of microtubules, but with a different structure. Okay? Now listen, 9 plus 0 structure. Okay? We call it 9 plus 0 because why? You have triplets. Triplets. That one triplets means 3, no? You have triplets of microtubules. Remember, three microtubules arranged in a ring. And there are no microtubules in the center. So we call that 9 plus 0 arrangement of microtubules where the cilium or the flagellum connects to the cell. Right? That is known as basal body. Okay, basal body. Got it? Okay, the basal body has 9 plus 0 arrangement with no microtubules in the center. Hari, now what we are going to do is we'll make a small note. Okay, we'll make a small note and so that you can complete your tute. Okay. Then shall we write, okay, methanolian, don't go into this side, don't go into this side. I want you to write here, I will write with you. Cilium or flagellum, that is the singular term, cilium or flagellum has 9 plus 2 structure. What do you mean by 9 plus 2 structure? Make a star mark, okay. Then you can write, what do you mean by 9 plus 2 structure? 2 doublets of microtubules arranged. Write with me, yeah. I don't. Therefore, you don't. I mean, I have to go ahead, no? So you won't get time to write. 9 doublets of microtubules arranged in a ring with 2 central single microtubules. Then next point we are going to write about the basal body. Another star mark can write down. Basal body anchors the cilium or flagellum to the cell. Next point, I'll go to the next page because my handwriting is big. You can manage in the same, you have to manage in the same space, huh? You have to manage in the same space. Composed of 9 triplets of microtubules arranged in a ring with no microtubules. Write with me, at the center. Okay, no, with no microtubules at the center. Within brackets write down 9 plus 0. Okay, 9 plus 0. Okay. Cilium or flagellum has 9 plus 2 structure, which is 9 doublets of microtubules arranged in a ring with 2 central single microtubules. Basal body anchors the cilium or flagellum to the cell, composed of 9 triplets of microtubules arranged in a ring with microtubules at the center. Clear? Okay. Then I want you to highlight in these diagrams. Can you highlight the names, or if you can highlight in Nathang, you can sort of circle them as basal body. Okay, then I want you to highlight plasma membrane, that is been covered by plasma membrane. මේවා හිතන්න ඕනේ නෑ මේ dianine nexim ඕවා ඕනේ නෑ ඔයගලන්ට. Okay, that's it. Then if you can highlight microtubule triplet, central microtubule doublet or whatever, right? You can highlight. Then most importantly, please write down this note underneath this. Okay. Any questions? Okay, then act as locomotor appendages. Okay, act as locomotor appendages. There, what is the function of this arrangement? It's how it's composed of. Actually, this basal body has a much more complicated structure. Scroll down, sorry. Okay, this basal body has a much more complicated structure than we learn here. Okay, when a cilium rotates, sorry, I mean a flagellum, sort of we say it's actually rotating, no? So sperm actually moves by rotating its flagellum. So for that, there's actually a motor-like structure, like something you see in a electrical motor wage. There's a cellular motor-like structure related to the basal body. We don't learn that here. So this arrangement of microtubules is there to provide a lot of support to the flagellum so that it's very stiff. It's not stiff means it's a very durable structure, so that if you take, okay, we'll talk about this when we are talking about the functions of cilia and flagella. Okay, anyone who's still writing this? Okay, come back to the resource. Okay, come back to the resource. What are the functions? Okay, what are the functions? They are given as locomotor appendages. Hurry, locomotor appendages it's given. So I want you to write down them. I'm going to write here. No, wait. So I want you to write down on the resource. If you don't like to, okay, right. This is from last week. Ne? If you don't write with a pen, that's fine. I want you to write down two things. Okay, two things. Act as locomotor appendages, flagella. I want to write down example. Okay, human sperm. Then cilia as a locomotive appendage. What organism do you know as cilia as locomotive appendage? There's some protist, the paramecium killer. There's a protist called paramecium. I'll show you a paramecium. He has a ring of cilia around him, so you can write that. Okay, paramecium has this sort of ring of cilia. Then the second one can move fluid over the surface. So this is the first function given in the resource. This is the first function, no? Act as locomotor appendage. Then for the second function, in front of it you can write down in the human respiratory system. In the human respiratory system. Okay, in the human respiratory system, you have cilia actually. So add by cilia in front of the second function that is given in the resource. Can you see here in this page? Okay, can move fluid over the surface. In the human respiratory system, by cilia can move fluid. Okay, I will explain this. Okay. Then the third function, cilia lining the oviducts help to move an egg towards the uterus. This I will explain. Okay, this I will explain with the software. Alright. Clear? Can I jump to my software? Alright. So you need to have a small idea about the female reproductive system, which is good to know always, Harry, because one thing, it's a big section in your biology syllabus, okay, the reproduction. If you take the reproduction lesson in your resource book, it's mainly about 20% male reproductive system and about 80% female reproductive system, okay. So if you generally in life also you need a lot of knowledge about the female system, okay. Anyway, so if you move on to the female reproductive system, if you remove some of these surface organs, you can see this is a cross sectional view, right? So okay, this is the rear view. We'll focus from the front, okay. So from the front, one half is missing, so that okay, so all these external organs you don't need. We are going to mainly focus on these internal organs. So in order to make it more simplified, with all this, without all these digestive system things, okay, so I'll go to a more simplified version like this, okay. Then what they say in the, what do you mean by this cilia help to move an egg in the oviduct? Now what do you mean by the oviduct, Hari? So this is the vagina. This is the vaginal canal that is connecting the internal male female organs to the outside through this opening. Hari? Then so that's the vagina. Vagina connects to what? This is where the baby develops, okay. That is called the uterus, right? So from the uterus you have your fallopian tubes, we call them oviducts. Usually now we don't, I mean, usually medical fields, doctors usually still use the fallopian tube, but now they are kind of removing that from your sort of textbooks. Gradually modern textbooks, they don't have that word much, okay. So this is the fallopian tube, okay. Then this is the female gonad, that is the ovary, this is the ovary, okay. So the egg is actually produced in the ovary. I can show you a cross section of the ovary like this, okay. So the ovary is this is how gradually an egg develops. Now listen, this egg, once this egg, so we say that is the production of the, or the, we can say follicle maturation is completed, the egg will be released into where? The fallopian tube. It will be released, but you can see there's a small gap also. So that gap, through that gap, the egg will go into the fallopian tube. So inside this fallopian tube, you have cilia. Inside this fallopian tube, you have cilia, and those cilia are going to slowly move with their rhythmic motion, slowly move the egg towards where? Towards the uterus. Slowly it is going to go along the uterus, sorry, fallopian tube towards the uterus, which is this, okay. So that's what they mean by help to collect the egg into the oviduct, okay. Clear? Then what did I mean by the respiratory system is something like this. Hurry, listen. What I meant by the respiratory system, if you take the respiratory system like this, you have a certain sort of a surface tissue layer, cell layer in your respiratory system. From here, so these are your lungs, no? From here in your lungs, this is the, we call them bronchi, may branching tubes, we call them bronchi. Then this is the trachea, you can feel your trachea here. Then in there, in those tubes, you have a layer of cells that have cilia, okay. So you know, into your respiratory system, you always breathe in so much waste, dust, no? Okay. So much dust, so much particulate matter, what are viruses, bacteria, fungi, pollen, and like there are carbon particles in the vehicle smoke, everything you breathe in, no? So it's going to get collected because you can't stop breathing, right? So it's going to get collected. So this cilia in your respiratory system is going to slowly beat, and they are going to move it slowly along this respiratory tubes until where? Until the common place that is between the respiratory system and the digestive system, which is what we call as kena. What do you call the common place? Common area to the digestive system as well as the respiratory system in the area, this area, that is the pharynx. Okay, so at the pharynx, what's going to happen is this all this dust that were coming up from the respiratory system is going to get swallowed, is going to come up here and it's going to down in the food tube. What is the food tube? That is your esophagus. So this esophagus is going to go where? Into your built-in acid bag in the stomach. That is the stomach where you have a lot of, very low pH HCl bag. Okay, so in that HCl bag, all this bacteria, all this particulate matter, viruses, fungi that you have breathed in into the respiratory system is going to get broken down. Okay, so if you think about it, you are not designed to or either cough or clear your throat and spit out. ඔගොල්ලෝ design කරලා නැහැ කෙල ගහන්න. You are not designed to spit your mucus. You are designed to eat your own mucus. Sorry, swallow your own mucus. That is the body designed. That is how the body designed. Okay, because you are spitting them out is a good way that you spread diseases. Okay, sure, you are not designed to spit them out. You are supposed to swallow them so that they will get broken down in your stomach. Okay, so you are designed to eat your mucus, and how that mucus with all the dirt and the particulate matter comes up is through the cilia that you have in this trachea. Okay, trachea and the respiratory tube system. Hurry. Okay, we'll come back to the tute. Any questions? Come back to the tute. Okay, so that is all about cilia and flagella. We can move on to the next structure that is given in your resource, that is centrioles. Okay, centrioles is very important when we are moving on to cell division. When the bacteria infect the esophagus, actually esophagus, the movement is very fast. Okay, and you will see the esophagus has a very several layers of cells to protect the surface of the esophagus, and they actually move down very fast because you have something called as peristaltic movements. Remember, rhythmic movements, I think you have in your O-level science resource book, textbook also you have. Okay, so because of this peristaltic movements, everything in the esophagus moves down fast. Okay, so there's no time for them to infect. Okay, so now we are going to talk about the next topic. You can put a subtopic here. I'll erase this. You can put a subtopic here as centrioles. Okay, put a subtopic here and read the resource. What is given in the resource? Centriole is made up of cylindrically arranged microtubules which are non-membrane bounded subcellular component present in animal cells. A lot of information is there. Alright, highlight non-membrane bound. Centrioles are not membrane bound, present only in animal cells. Okay, then each centriole is composed of 9 sets of triplet microtubules arranged in a ring. Alright, so now you can see in this diagram. So you can see the relative location in a cell as well. So if you take a cell like this, you are going to have a pair of centrioles in a structure that we call as a centrosome. So in a centrosome, we have 2 centrioles. What are centrioles? Centrioles are these. Okay, so centrioles actually have what? They have 9 triplet microtubules arranged in a ring. So I think you can, if you zoom in, you can see 1, 2, 3, 1, 2, 3. There are 9 triplet microtubules arranged, so that is what we call as a centriole. Can you highlight? Each centriole is composed of 9 sets of triplet microtubules arranged in a ring. So it also has, we can say, 9 plus 0 structure. Okay, it also has 9 plus 0 structure. So there is a single centriole if you take what we call as a centrosome, you have two centrioles arranged perpendicular to each other. So you can see this is 90 degrees. You have two centrioles arranged 9 at 90 degrees. So that's what it says in the resource. Read. A pair of centrioles which are arranged perpendicular to each other are located in a region called centrosome. So actually, the pair of centrioles that are arranged perpendicularly is actually the centrosome. But we also call the location in the cell where you have the centrosome as the centrosome region also. Got it? The centrosome area is the centrosome region. Where you find the centrosome is the centrosome region. So centrosome is actually two centrioles arranged perpendicular to each other. Okay. So that region is also called as the centrosome. Got it? So what I want you to remember now, something you should be making a small connection here. Neither. So this has 9 triplets of microtubules arranged in a ring. What else did we just learned that has 9 triplets of microtubules arranged in a ring? What else? What else did we just learn? Basal body, no? Okay, basal body. So you can say basal body as well as the centrioles have similar structure. Okay, so we'll write here. I want you to put a star mark. Okay, put a star mark here. Centrioles share the same 9 plus 0 arrangement of microtubules microtubules as basal body of cilia and flagella, as the basal body of cilia and flagella. Okay. Centrioles share the same 9 plus 0 arrangement of microtubules as the basal body of cilia and flagella. Okay. So when I'm like writing in a corner like this, sometimes even this small my name is actually a waste of space again. So I have seen tudes from in other classes where there's a massive watermark across the full page, right? So that is kind of distracting. No, the student is not able to properly work on the tude page. So that's why I always try to keep even my name very minimum, so that it's just indicating it's my tude. But this is not my knowledge, no, this is not the diagrams that I designed. So actually I am not at liberty to put a massive watermark. Okay, so I try to keep it minimum. Even this is actually a bit too big again now I think of it. So because it's space I could have used to write. Okay, anyway, centrioles share the same 9 plus 0 arrangement of microtubules as basal body of cilia. Okay. Right. So you can see here an electron micrograph. What is the size given? 0.25 micrometer. So you can see how small it is. Can anyone tell me what sort of electron microscope did you take this image? What sort of electron microscope you used to take this image? You now know two types of electron microscopes. What do you think is the electron microscope we used here? Yeah, it's a transmission. No? Why do you say transmission? Because you can see the cross-sectional view. No, Metin? You can almost see the microtubules. You can see the microtubules. If you are seeing a cross-section, that means you are using something that uses electrons going across. Okay, so this is a TEM. Usually the standard electron microscope is actually the transmission electron microscope, because scanning electron microscope is a bit more difficult to use and a bit more expensive, and you actually have very limited applications. You don't need scanning electron microscope images usually. You usually need cross-sectional inner views. Okay, so that is it. Then functions of the centrioles in the resource book: produces aster and spindle in cell division. So just highlight it. You will understand very soon what do you mean by aster as well as the spindle. Okay, so actually aster is part of the spindle also. You will understand, okay? Then we move on to central vacuole, okay? Then we move on to central vacuole. Actually, even though we only, the topic there is central vacuole, I want to talk about 3 types of vacuoles that you encounter here and there in your syllabus. I want to talk about 3 types of vacuoles. Vacuoles කියලා කියන්නේ they are mainly like a water tank. If you take the central vacuole, it's like a water tank. The other vacuoles, there are vacuoles that act as a lunch box. There are vacuoles that act as a lunch box to a cell, and there are vacuoles that act as like a small kidney. Okay, so vacuoles are very diverse in their functions. So therefore, let's make a small classification like this. So vacuoles are usually membrane bounded structures that you find in the cell. You can divide vacuoles into 3 types, okay? Based on their structure and function, you have 3 types of vacuoles, okay? Okay, 3 types of vacuoles. First one is the one I told you, it's like a lunch box. Those are called as food vacuoles. Food vacuole is like a lunch box, okay? So food vacuoles are involved in something called as the cell eating. What do you call the cell eating? Taking in some food in so that the cell can digest it. What do you call that? Anyone? What do you call when a cell is eating? I think we did this. I had a diagram when we were talking about yes. Maybe make. Now, balla kiyanna ba. This is how a cell eats. Yes. Anyone? What do you mean? What do you call when a cell is taking in food particles? Yes, yes, yes. Go. Yes. What do you call when a cell is taking in food? Metin, it's given on the board. No, no. Not autolysis. Don't get confused. It is phagocytosis. Simply phagocytosis. Phagocytosis is when a cell eats. Okay. That means the cell is taking in food particles. So you can see, this is a food vacuole. That means it's a vacuole, that is a membrane bound package that is containing some food inside. Okay. So that is the one type of vacuole that you need to know. Okay. So we can say food vacuoles involved in phagocytosis. So in your body there are cells that eat other cells. We have cells called macrophages that are very important in your immunity. So when you have some bacteria in your body cells, your macrophages, which is a type of actually you can say white blood cell, they are going to go and eat it. They are going to go and eat it. So for that the process of phagocytosis helps. Okay. So involved in phagocytosis, you can say helps with digestion. Okay. So that is the function of food vacuole. The second type of vacuole you need to know in your syllabus is, so we make a madillium, it is the central vacuole. Central vacuole. As the name says, the central vacuoles are usually very big. If you take a plant cell, a big portion of the plant cell is actually occupied by the central vacuole, not the nucleus or anything. Okay, the biggest thing in the plant cell usually is a central vacuole. Okay, so central vacuole has actually some names you need to know. If you take a central vacuole, it is a, I told you it's a membrane bound structure. So it has a single plasma membrane, similar to a plasma membrane, there's a membrane. Then inside you have a fluid. Inside this you have a fluid. I told you the central vacuole is like a water tank. Okay, so inside you have a fluid and the membrane, it's membrane bounded. So we call this the membrane of a central vacuole, we call it tonoplast, and the inner fluid we call it as, can anyone? Cell sap. Hari? Cell sap. Then I want you to write down a list of things that is found in the cell sap Actually, your cell sap contains, usually I told you it's like a water tank, right? So mainly it's a water store, okay? So it contains water. Dissolved in water, you have some other things needed for the plant's metabolism. Ions, right? Ions are important for certain signals to travel. Ions such as potassium, K plus and Cl minus. And then certain pigments, colors. You have seen different plant leaves can have different colors, nice colors. Okay, so there are like purplish colors. Purplish, that reddish blue color is given by a pigment called anthocyanin that you need to know. Anthocyanin. Also, so other than water, ions, pigments, then you can have some sugars as well. Plant sugars, sucrose, whatever you can have in this cell sap that is found inside the central vacuole. Okay. Finally, you have a type of vacuole that is kind of acting like a kidney. A kidney in our body wage. If you take a unicellular small organism such as a euglena, paramecium, protists, okay, they have a vacuole that is kind of acting like a kidney. What do you mean by, what is the purpose of a kidney? A kidney regulates the amount of water and certain ions that you have in your body, no? Sodium, chlorine, potassium. Those kind of iron is balanced by your kidneys. So, since our kidney is a big, very complicated structure, these cells have a very simplified version of it that is done by a type of vacuole called contractile. I'll show you a contractile vacuole. Contractile vacuole. So, their main functions are, yes, what are the main functions of a contractile vacuole? Osmoregulation. Hari, what do you mean by osmoregulation? Is the maintenance of water balance. So, you can say maintain water and electrolyte. Electrolyte means these sort of charged ions, potassium, sodium plus, potassium plus, Cl minus, water and ion balance. Electrolyte, sorry, water and electrolyte balance. That is the function of a contractile vacuole. Hari, can you write down here in this space? Can you write down in this space the three types of vacuoles together with their functions? Draw a small central vacuole and write down its different structures as well. Okay. Okay. We have to quickly finish our cellular structure so that we can move on to cell division. Okay. What about animal cell vacuoles? What do you mean? Animal cells, and usually animal cells don't have vacuoles. Okay. Except for food vacuoles found in certain cells eating other cells. Tonoplast is the membrane of the vacuole. Yes. Central vacuoles are only. Yeah. Can you add here? That's good. That's a good point to add. Okay. So central vacuoles, we will add it. Okay. Central vacuoles. I don't have much space here. Please add here underneath central vacuoles only found in plant cells. That's a good point. Okay. Only found in plant cells. You can add here for the central vacuole. Contractile vacuoles you can usually found in your certain protists, right? Certain protists you can find. And food vacuoles you can find in even in your body cells, such as some white blood cells that are able to eat other cells. Animal cells very rarely have. If you have vacuoles, that's actually a disease, bit of a disease condition. There are certain diseases that cause vacuoles in your brain. Okay, those are what we call as prion diseases. That is something you have in your microbiology. Okay, you have vacuoles forming in your brain due to a yes. CJD it's a Creutzfeldt-Jakob disease, which is a disease that can cause sort of vacuoles in your brain. Okay, so that you gradually lose your mind actually. Okay. Anyway, no need to focus on that. So we'll come to the resource and we'll read what is there under the resource. Okay. So tonoplast is the membrane of the central vacuole. Cell sac is the inner fluid. Okay. So let's read. Central vacuole is a large structure bound by tonoplast filled with a liquid called cell sac. Highlight. It's a large structure bound by tonoplast filled with the liquid called cell sac. Composition of the cell sac differs from the cytosol. Then highlight what are the things. Water, ions such as potassium and chloride, sometimes water soluble colored pigments such as anthocyanin. Okay, anthocyanin. Then of course you can have sugars as well. So functions of vacuole. So actually I want you to write down in front of each of these functions that are given done by which vacuole. Okay. So the first function is given as store water and other materials such as iron, sugars, ions and pigments. Write in the resource in front of that. That is done by Kiana, who stores sugars, ions and pigments. Central vacuole. Central vacuole. Write in front. Central vacuole. Then maintain water balance of the cell or osmoregulation. That is mainly by who? In front of the second function, write down. Contractile. Write down contractile vacuole. Also, you can put a comma. A bit of osmoregulation, maintain water balance is done by central vacuole as well. So write down both. Okay, maintain water balance in front of it. Write down central as well as contractile vacuole. Okay. Third one, gives turgidity and support to the cell. I will explain. Okay. Then in front of it, write down give turgidity and support. That is done by central vacuole. Okay. Then that is done by central vacuole. The next function, produce colors in some plants with sap pigments. That is again central vacuole. Finally, helps in digestion. That is what? Food vacuole. That is food vacuole. Got it? That is food vacuole. Helps in digestion. I will explain what do you mean by turgidity. What do you mean by turgidity when I am explaining the cell wall. Okay, clear? So did you highlight the composition of the cell sap? Yes, water, ions such as potassium chloride, watercolor pigments. It can contain sugars as well. So we wrote it here. No? So that's all you need to know. Okay. Okay, we are going to move on to the cell wall. So now everything we have talked about up to here are things that you can find inside the cell. Even if you take your cilia and flagella, we consider them intracellular or found within the cell. Why do we call them as intracellular? What is the definition? If something qualifies to be within the cell, what is the boundary? Then if something is within Sri Lanka, we have international boundaries there. So if something is found within a cell, if you want to consider something as intracellular, what is the requirement? It should be inside the plasma membrane. It should be inside the plasma membrane. So anything that is inside the plasma membrane is intracellular. So even the cilia and flagella, even though they are projections out of the cell, they are still bound by the plasma membrane. So they are intracellular. No? So we have done all the intracellular things, say cytoskeleton, mitochondria, chloroplast, lysosomes, peroxisomes, all things, nucleus, everything is done. Now we are going to move on to structures that are belonging to the cell but outside the plasma membrane. Okay, so therefore extracellular. Okay, let's read. Okay, come to page number 37. Extracellular components, cell wall. Extracellular components, cell wall. So this is the image of the cell wall that I want you to keep an eye on as we are reading, right? Please have your resource book as well as the diagram tube open so that while you are reading you can look at the image. Okay, so we will see what do you mean by cell wall. Highlight: cell wall is an extracellular structure of plant cells. Okay, animal cells do not have cell walls. Highlight: animal cells do not have cell walls. Usually animal cells do not have cell walls. Okay. Prokaryotes, fungi and some protists have a thin and flexible cell wall. Alright. Prokaryotes, fungi and some protists have a thin and flexible cell wall. Prokaryotes, what do you mean by prokaryotes? Usually you have bacteria. Fungi, can anyone tell me what is the fungus cell wall composition? What is the fungal cell wall composed of? Yes, chitin. Good. Okay. Then protista. Protista, if you take protista, there are algae, no? Algae. Green, brown, or algae are there. So those algae have a cell wall. Usually it is kind of cellulose based. Okay. Similar, very similar to a plant cell wall. Okay. Thin and flexible cell wall. Chemical composition of the cell wall greatly varies from species to species, even from one cell type to the other within the same species. Okay, because depending on the location, if you take a plant, there are so many variety of different cell walls because of the different cell types. There are cells that are designed to do photosynthesis, so they have usually thin walls. There are cells that are designed to give support to the plant. They usually have a thick cell walls. So that depending on their function, they have different structure of cell walls. Okay. In plants, cell wall is generally made up of cellulose, pectin and hemicellulose. Lignin and suberin are also found. Can you highlight the composition of the plant cell wall? Highlight the composition of the plant cell wall. Cellulose, pectin and hemicellulose. Okay. Lignin and suberin are also found. That is, they are kind of like secondarily. Lignin and suberin is kind of there secondarily because they are mainly produced when the cell requires a bit more support. Okay. Plants generate two types of cell walls, primary and secondary walls. Young cells first secrete the primary cell wall. It is the wall laid down during plant cell division. Okay. So highlight: young cells first secrete the primary cell wall. You first, whenever a young plant cell has been generated, you have a primary cell wall. Primary you get into your mind, your small chutikale again. So primary cell wall, the cell wall you get first. This is the primary cell wall. Okay. We'll go through the resource, then I'll explain this. Okay. Just outside the primary cell wall, there is a thin. Okay. Amazing. There is a thin layer, middle lamella, which is rich in a sticky polysaccharide called pectin. Pectin is actually a metal mixed carbohydrate. So there's a metal component in the carbohydrate that we call as pectin. It's a polysaccharide. So usually magnesium and calcium salt of pectate you have. So it's a existing as a salt. Now, for example, so if the example I like to use is, then if you take two plant cells, sorry, now listen. If you take two plant cells that are neighbors, neighboring plant cells, two plant cells, if you take, sorry, they usually have this cell wall. So they are like the bricks of a wall, ne? Wall, I mean bricks of a wall, gadol again bricks. So if you just have plant cells that are just just connected to each other, or similar to just bricks, you just place bricks on top of each other, is it going to be a very strong wall? No. You require some sort of a material between these cells to hold them together. So you get a nice plant tissue, no? So if you want to build a strong wall, you need a cement. So the cement in your plant cells, between your plant cells, is actually your calcium and magnesium pectate. So you take pectin and you infuse it with metal ions such as calcium and magnesium, you get magnesium and calcium pectate. So it's like you take cement from the so you take cement powder and you mix it with water, sand, and then you get a mortar, no? Badam mortar. So that's what you have in the middle of this plant cell wall. Sorry, plants, yes, in between plant cell walls, like a sandwich. Okay, so you can see here, this is the middle lamella. You can see here also, this is the plasma membrane. So outside the plasma membrane, you have the primary cell wall. Then outside that, you have the cement, which is the middle lamella. Okay, keep reading. Middle lamella glues the adjacent cell together. Okay, did you highlight the composition? Middle lamella rich in a sticky polysaccharide called pectin. Rich in a sticky polysaccharide called pectin. Rich in a sticky polysaccharide called pectin. Composition is calcium and magnesium pectate. Highlight that. Composition is calcium and magnesium pectate. Okay. Then okay, due to the deposition of hardening substances in the primary cell wall, a secondary cell wall is generated secondarily. Highlight. Deposition of hardening substances in the primary cell wall. Nice. So this is your primary cell wall, which is a nice structure. Okay. So you have actually they have given the enzyme that is making cellulose called cellulose synthase. Okay. So he is making constantly new cellulose, so that you have a crisscrossing cellulose fibers. If you take zoom into your cloth, you can see the fibers. In the cell wall, you have cellulose fibers that are sort of crisscrossing, and so you have a bit of pectin in between. You can have hemicellulose in between cellulose fiber. So it's a nice structure. The most important thing, remember, the most important thing about the primary cell wall is it's permeable. That means things can go across it. Water and other minerals, plant nutrients can go across this the primary cell wall. It's a permeable structure, so that anything that is coming outside can go through the plant cell wall, primary cell wall, and then reach the plasma membrane, and so that materials can exchange. That is the feature of primary cell wall I want to remember. So can you see next sentence in the resource? Read. Primary cell wall is permeable, relatively thin, flexible, and mostly composed of cellulose fibers deposited on the middle lamella. Yeah, so you can say this is the middle lamella now. You can say this is the primary cell wall. It is usually thinner and it's highlight permeable. Very, very important. Highlight, underline with a red pen even if you want. It's a permeable structure. Hurry, permeable structure. Then water can move freely through the free spaces. Yes, highlight with a different color. Water can move freely. Okay. Now we move on to the secondary cell wall. Secondary cell wall lies between plasma membrane and primary cell wall. It contains several layers of hard materials forming a rigid structure. Okay, so now not after some time, if you are designed to be, if you, by fate, you are a plant cell who is going to have a secondary cell wall. That means you have a death or a death sentence. Okay, so usually secondary cell wall is generated in cells that are going to become supportive cells, right? That are designed to provide support to the plant or the tree. So if you are a cell like that, from your birth, you have a death sentence. That means you are going to die soon. That is because why? If you come into the secondary cell wall, and okay, now this is, we are going to consider primary cell wall like this. Okay, then inner to the primary cell wall, you are going to generate a new cell wall that also has cellulose. But most importantly, can you see, there's a new man, that is lignin. Can you see lignin all over these spaces? Maybe can you see lignin? That lignin is going to have a massive effect. Can you read? It contains several layers of hard materials forming a rigid structure. In addition to cellulose, impermeable substances such as lignin and suberin are also incorporated. So you can say lignin, meaning you can write lignin plus suberin. You can write lignin plus suberin is also there. Suberin is also there. Lignin plus suberin. Lignin as well as suberin is there. Okay, in the secondary cell wall. Lignin cement anchors cellulose fibers together, providing hard and rigid matrix. Highlight. Lignin anchors the cellulose fibers together, providing hard and a rigid matrix. Okay, so that is, this is a much stronger wall than the primary cell wall. So you can see suberin and lignin sort of in between cellulose fibers in the secondary cell wall, forming a hard and rigid matrix, giving the cell wall extra support. Okay, jump to the next page, page number 38. So, cell wall has pits through which cytoplasm adjoining cells join through plasmodesmata. I will explain what is plasmodesmata in the next section, that is cell junctions. Okay, right. Then I told you I'll explain a concept. But before that, I want a few things to be written here. So plant cell wall, the topic is already given. No, I want to write down a few things here. Let's write. I want to write down a few things here. Actually, we are going to write small few points about the primary cell wall as well as the secondary cell wall. So under, write down a topic, primary cell wall. Okay, primary cell wall. Write down what are the features. It is permeable, thin, flexible, mainly composed of cellulose deposited on middle lamella. Permeable, thin, flexible, mainly composed of cellulose deposited on the middle lamella. That is primary cell wall. Okay. Then on this side you can write down about secondary cell wall, which is like a death sentence. Why the secondary cell wall is like a death sentence? Once you get suberin and this lignin, all the things, water and other things cannot go through the secondary cell wall. So that means you are cut off as a plant cell, you are cut off from the rest of the plant. Therefore, you can't survive, you are going to die. Then write down on the secondary cell wall. You can see it is found inner to the primary cell wall. Found inner to the primary cell wall. This is something you forget usually. So that means don't forget. Okay, primary cell wall is usually located outer to the secondary cell wall. Primary cell wall is located outer. So in between your primary and plasma membrane, you have the secondary cell wall. Okay. So that's what we are going to write here. Formed between So like a sandwich එකක් වගේ primary cell wall and plasma membrane formed between primary cell wall and plasma membrane contain impermeable substances such as lignin, suberin, anchoring cellulose fibers together. Anchoring cellulose fibers together. Okay, quickly write this down. Comparison between primary cell wall and secondary cell wall. Okay, quickly write this down. Okay, right, huh? Right, then I want to move on to functions of cell wall. Can you read? Protection and support. Yes, of course, it provides the it maintains the shape of the plant cell and helps to maintain the shape of the plant itself, because it's how usually if you take chuti plants, they are brought, kept straight by the support of the cell wall. Okay, allows development of turgidity when water enters the cell. Now this is a concept that I want you to understand. This is something we are going to එපා වෙනකන් ඉගෙන ගන්න concept then we have in plant form and function. හරි. So if you take a plant cell, I told you more than the nucleus even, the dominating structure in a plant cell is actually a central vacuole. Remember, tadi central vacuole is there. Actually, your nucleus and other things are found on the side. So, this is the nucleus and this is the endoplasmic reticulum. You have mitochondria, chloroplast. All the other things are actually found on a side. The main thing you have, the dominating structure is actually the central vacuole. So, now listen, what's going to happen is, we are going to apply Newton's third law. Take apart why do you need Newton's third law? It's like this. Right? When water comes in through the primary cell wall, so now we are talking about a living cell. When water comes in, this vacuole is going to get bigger. Okay, so when the vacuole gets bigger, the entire cell cytoplasm, that means the cytoplasm is what? Cytoplasm is the gel-like thing. No? So the entire cell cytoplasm is also going to get bigger. So now when it's going to get bigger, your cell wall is a bit of a rigid structure, or it is composed of cellulose. So what's going to happen is this cytosol is going to press on the cell wall. Okay, this is going to press on the cell wall. Push එකක්. So you know every action has a equal and opposite reaction. What is it? Newton's which law? Is it third law? Okay. Right. I gave up learning physics a long time ago now. Okay. Right. So when the cytoplasm is pushing on the cell wall pushes back. Reaction force. Okay. So therefore, inside the plant cell, there's going to be like a pressure produced because every side it's being pushed. From every side it is being pushed. Therefore, the plant cell is going to be under pressure. That sort of a pressurized condition we call as turgidity. Turgidity. Okay. So allows development of turgidity when water enters the cell. That means it allows the plant to get firm. The cells get under pressure. That means under pressure means what? You are firm. Okay. So prevents bursting during turgidity. That means because you have a plant cell wall preventing a plant from bursting. If you take one of your cells, they can easily, like a balloon, they can burst. Because why? You only have the plasma membrane surrounding you. So through the plasma membrane, you can burst. Limits and control cell growth. Yes, this acts like a wall. No, so the plant cell has a cell growth limitation due to the cell wall. Highlight, okay, limits and control cell growth. Did you highlight allows development of turgidity and prevents bursting during turgidity? Component of apoplast pathway. Okay, I'll explain the apoplast pathway when I'm explaining the plasmodesmata as well. Okay, keep that just for now. Apoplast pathway, highlight apoplast pathway. Maintain cell shape. Yes, maintain cell shape and hold the plant up against the force of gravity. Highlight that also. Hold the plant up against the force of gravity. That is again something done by this pressurized plant cells. So when the plant cell is pressurized, that means it has a lot of strength, stiff. So when you have like a set of this kind of cells, you can have a lot of tension. So if you take like a leaf, then we have a leaf එකක් ගත්තොත්, right? This leaf is connected to the plant at a petiole. Petiole කියලා කියන්නේ this sort of a connecting. So this is the plant stem. The leaf is connected by this. So imagine the cells found in this petiole are turgid, full of water. That means this leaf is going to nicely stay up like this. But if they lose their turgidity, what's going to happen? It's going to droop down. Okay, that's what you mean by turgidity. Okay, then we can move on to cell junctions. Okay, cell junctions. Any questions from primary cell wall and secondary cell wall? Everyone wrote this down, ne? People not sleeping. I know it's a bit raining, but what to do, Harry? Okay. Alright, so these days I have talked about this. Try to watch some good movies. So this is the only free time you are going to get for another 2 years. Watch some good movies, watch some good TV shows without scrolling on the phone. Sorry, that's a very bad skill if you get addicted to it. That is going to ruin your attention, your ability to concentrate, everything. Okay, so watch some good movies. That's some sort of that will make you use your brain a bit. Okay, so right now watching a 3-hour movie is more difficult than actually, because you get bored, no? Because without the instant let's say the ending of maybe a YouTube short or a TikTok or something, you should very fast you get bored. So that is something you have to improve. Otherwise studying can get difficult, particularly if you are doing some physics problem. Within 1 minute, if you don't get the solution, you tend to give up right now. I have seen that among students. So that is not something you should, because you should be able to focus, give your maximum for extended duration, because the 2-hour physics paper MCQ is 2 hours, right? So you have to have your maximum performance for entire 2 hours. Otherwise, if you run out of focus after 1 minute, That's not good. So the best way to develop it right now is, yes, do some questions and try to focus when you are studying. But at the same time, watch some movies and TV shows that require you are focusing. Otherwise, you don't understand. Nolan movies, same Christopher Nolan movies, same. Those things you have to pay attention. Otherwise, you lose what's happening. Okay, so try to watch some of those at the same time you are studying. Okay, do some sport. If you are playing instruments, practice those a little bit these times. You will get a lot of opportunities to show your skills when you go to university. Right? So develop some of your skills so in this time period, because they are good for brain stimulation. Okay, so try to do those other things as well. Anything but social media and this scrolling. Okay, anything is better. Physical activity, aesthetics. Okay, whatever. Okay, right. Cell junctions, let's read. Okay, so we have to complete this set of cell junctions. So I'll erase this. We have actually 3 types of cell junctions together with a special type of a cellular connection that we call as plasmodesmata. Okay. Okay, let's read. Cell junctions, there's a definition. Neighboring cells join, interact and communicate via sites of direct physical contact. Okay. Then highlight structure at which cytoplasm of adjoining cells are joined. Highlight structure at which cytoplasms of adjoining cells are joined. That is a cell junction. So in your syllabus, they connect the internal chemical environment of adjacent cells. There are 3 types of cell junctions present in animal cells. Can you highlight? These 3 are only present in animal cells. These 3 cell junctions are only present in animal cells. Break. I'll give a break. Let's finish cell junctions and ECM, extracellular matrix. Then you take a break and we'll start cell cycle. Right? That's good, no? That's what time. I'll give a break at 8:10. Okay. Okay. Cell junctions. So these type of cell junctions, that's what we are going to talk about. That's tight junctions, plasmodesmata, and your gap junctions are only found in animal cells. Okay, I want you to remember that. That's something they can ask in MCQs. These 3 cell junctions are only found in animal cells, not in plant cells. Okay, plant cells, there's a special type of cell junction that is we are going to learn additionally. Okay, so first cell junction, tight junctions. Read, there's a definition. Okay, so I think new people, you don't know this. I want you to have a dedicated color for definitions, either dedicated color for definitions, so that when you open a page of the resource book, you immediately see the definition first. Because what do you mean by definition? It's something they can ask you. Okay, what is a tight junction? So if they ask you in a structured essay paper, you have to write exactly this. You have to write exactly this. Okay, otherwise there's a chance you might lose marks. Okay, so tight junctions connect the plasma membranes of adjacent cells tightly by special proteins forming continuous seals. Okay, forming continuous seals. Highlight, that's a definition. Then underline some of the keywords. What do you mean by keywords? That means these are the things that they check for. So when you are writing a definition like thing, you definitely cannot miss keywords. What are the keywords? Connect plasma membrane by connect plasma membranes forming continuous seal. It's like actually a watertight seal. Okay. Prevent leakage of extracellular fluid through the intercellular space. So you can understand your tight junction is actually a watertight seal. A watertight seal, right? That is going to make, cells will not be able to, so water will not leak between the gaps of cells, right? Water will not leak between gaps of cells. For example, if you go to swim in the pool, are you going to get filled with water in your body? Are you going to get filled up with water? No. That is because your skin epithelial cells, so your skin epithelium has plate-like cells actually, plates wage cells, right? Your skin epithelial cells are actually if you zoom in, they are connected by special proteins in between each other, so that any liquid that you have outside is not able to go through that. That is what we call as a tight junction. You can see methane sealing strands. So this plasma membrane, and this plasma membrane is connected by special proteins, forming a watertight seal. Okay, watertight seal. So let's write down here මෙන්න මෙතන in front of the tight junction. So can you write down the topic tight junction here? So label this junction, it's already given actually, so ලොකුවට ලියන්න, but in your handwriting, tight junctions. Then write down some points here. You can say connect plasma membranes, forming a continuous seal preventing leakage. It's given as the example is given as skin epithelium, that means your skin surface layer. Example, skin epithelium. Okay, write down in front of the tight junction. Connect plasma membranes forming continuous seal preventing leakage. Okay, connect plasma membranes forming a continuous seal preventing leakages found in your skin epithelium. Okay. Next, come to the resource book, back to the resource book. Second one, desmosomes or anchor junctions. Desmosomes or anchor junctions mechanically attach the cytoskeletons of adjoining cells by intermediate filaments for strong binding. Okay. Definition, they can ask you what is a desmosome or anchor junction. Then there are keywords, no? Attach the cytoskeleton by intermediate filaments. Okay. What do you mean by an intermediate filament? So this is an anchor junction. You can see a electron micrograph also. So this is one cell. This is one cell. Usually could be a muscle. Usually muscle cells are connected like this. In between them you have your desmosome or anchor junction. So here also you can see this is your actual desmosome that is composed of very strong cables or intermediate filaments, and this is actually your what? You know this. What are these components? These are actually your inner cellular skeleton. Cell skeleton is known as what? What is the name of the cell? Skeleton of the cell? Okay. What is the name of the skeleton of the cell? Cytoskeleton. Okay. So you can see cytoskeleton again. Like imagine like they have taken the cell not by the plasma membrane, but you have gotten hold of its skeleton and you are connecting it with another skeleton of another cell. So imagine how strong they are. So desmosomes or anchor junctions are the strongest junctions. They are found in places where you have a lot of mechanical tension, such as inner skeletal muscles, right? You have to lift a lot of weight. Okay. So you can say here, you can put the topic anchor junction. The name desmosome is given here already. So you can say anchor junction Then write here attached cytoskeleton by intermediate filaments. Attached cytoskeletons by intermediate filaments. By intermediate filaments. Okay, example, muscle tissue. Okay. Okay. What are intermediate filaments? You know this name. What are intermediate filaments? I think it's a component of the cytoskeleton. Can you see? It's like a cable, no? Composed of several types of protein. It could be composed of several types of protein. Keratin is a common example. Okay, it's composed of several types of protein. Options are there. Okay, but usually keratin very strong. Okay, very strong. Hurry. Okay, done. Attached cytoskeleton by intermediate filaments found in muscle tissue. Next, final cell junction found in animal cells. Okay, gap junctions or communicating junctions. Let's read. Provide cytoplasmic channels from one cell to the adjacent cell. Highlight cytoplasmic channels from one cell to the adjacent cell. Can you see the diagram, Methi? It's like a series of doors or openings. Windows from one cell to the other. It's a series of windows. Okay, so you can see this is one cell, this is the other cell, and you have small openings forming channels. No, these channels are what we call as the gap junctions or communicating junctions. Okay, it's like between your house and the other house, your neighbor's house, you have a window that you can directly exchange food, shout. Okay, ever. Gap junctions consist of special membrane proteins that surround a pore through which ions, sugars, and amino acids may pass. They allow signal and material exchange between adjacent cells through direct connection. So, it's a direct connection, okay, between your house and your neighbor's house. You have a direct opening that you can use for material as well as signal. So, you can exchange food or you can give them a message. Okay. So, both things can happen. They are usually found in tissues where you need a lot of coordination, where you need a lot of message to be sent everywhere in the tissue. Okay. So, that is what we call as a gap junction or a communicating junction. So, we can say it's a gap junction. Can you label it? Gap slash communicating gap slash communicating junction. Okay. Then you can draw a small diagram also, polydiagram you can draw. These are the two plasma membranes. These are the two plasma membranes. Then you can draw some proteins. They are arranged like a tube. You can see, and they extend to the other cell, the other side. Okay. So they have this sort of an arrangement. Okay. Okay. They have this sort of an arrangement with a. You can transport things through. So you can call this as the pore. Okay, pore. And then what are the things that are going through this? sugars, ions, amino acids. Okay, those are things that are going to go. And some examples where do you have your usually communicating junctions where you need a lot of communication. Okay, for example, when you are a ball of cells, without any arms, legs, heart, lungs, right? When you are a ball of cells, you had a lot of gap junctions that helped you to that ball of cells to have a nice development together, to coordinate the development. Okay, so animal embryo and heart muscle, because your heart should beat as one, no? So they should know if your atria are beating, your ventricles should know, okay, I'm next. Your heart should have an overall nice coordination. If the heart doesn't know what's happening, you are going to have an abnormal heart rate and your heart is not properly working anymore. Right? Then I told you I'll explain a little bit about this plasmodesmata. Okay, we'll discuss about plasmodesmata and we'll take a break. Okay. Page number 38, final small description. We'll take a break after discussing plasmodesma, ECM after the break. Okay. Microscopic channels which run through plant cell walls, highlight, they are microscopic channels which run through plant cell walls. They are cytoplasmic living connections between cytoplasm of adjacent cells. They are membrane line channels filled with cytoplasm. If you take two plant cells, I want you to actually draw it here. But because I don't have much space, I'll go to the whiteboard. Okay, so otherwise I won't be able to explain it nicely. Okay, so if you take two plant cells like this, they are bound by the cell wall. No? They are bound by the cell wall. So it's like your house, your land, the land of where your house is located, and your neighbor's house. Okay, your neighbor's land, actually, the neighbor's land. So you have a wall separating your land from your neighbor's land. Okay, so a plasmodesmata is actually like a hole in this wall. It's like a hole. Okay, so that the plasmodesmata, your cell wall itself is having a opening, and the other thing is, so if you take the inside, so you have the cell wall, then one is inner to the cell wall, you have your plasma membrane, no? The plasma membrane of the plant cell. So actually the plasma membrane of one cell is actually go through the plasmodesmata into the next cell. Okay, so this cell is independent. So this cell has its own vacuole, this cell has its own vacuole, and he has his own nucleus, and he has his math has his own nucleus, and he has his own mitochondria, blah blah, all the other organelles are there. But the special thing is, there's this channel called the plasmodesmata, which is membrane lined. That means it is, the plasma membrane is actually going through the plasmodesmata into the other cell. So it's a membrane lined. And what do you have in the middle? What is the filler? It's actually cytoplasm. No, it is still cytoplasm that you have, that is flowing through the plasmodesmata. So cytoplasm is found everywhere else in the cell, everywhere else in the cell, as well as it is going through the plasmodesmata as well. This is how the plasmodesmata looks like. Okay, so I want you to draw this in that space that you have under the gap junction. So label it as plasma membrane. This as plasma membrane. Okay, then I want you to write down the three features of the plasmodesmata. One, they are membrane lined. Of course, they are membrane lined. You can see, right, if you take something like the gap junction or communicating junction, they didn't have a membrane, no? They are just composed of proteins. Okay. And the other thing, because it is filled with cytoplasm, it's a living connection. It's a living connection. It's part of living cell. Then filled with cytoplasm. Okay. Filled with cytoplasm. Filled with cytoplasm. Okay. These are the features. I want you to draw this. No need all these vacuum organelles and other things. Right? I do no need to draw all this, but I just want you to have the label the cell wall also. Sorry. Okay. Label the cell wall also. Label the cell wall. And then label the plasma membrane. Write down the three fundamental features. That is, they are membrane lined, they are living, as well as they are filled with cytoplasm. Okay. Any questions? Any questions? I want you to write it here. Okay. At the end of there's a space. Okay. Can you draw it here in your tute? There's some space. Okay. So people who got the tute today, don't worry. You will get it delivered. Then you can watch this recording. Or if you are taking it down somewhere, you can then complete it. Okay. No need to think about it. Okay. Okay. All right. Then after the break, we'll discuss about the extracellular matrix, and we are going to move on to cell division, mitosis, together with the cell cycle discussion. Okay. So quickly come back. Okay. Any questions? Any questions you want to clarify? Taggalan, taggal neve, hemita hande. Okay. White color gellila hakene. Okay. Break. How long do you need? Join by Ken. So show the diagram again. Okay, sure. 8:15 with the end. Okay. Workout over 8:20. Okay. Okay, go and come back quickly. Okay. You can get the 3D software once you go to into medicine. No need. Okay. I didn't have this when I was doing A levels. So you can. It's very useful when you are in medicine or something that is related to studying anatomy. But otherwise, a bit not necessary in your A level life. Okay. It's called 3D anatomy or something. Yes. Okay. 4 people. Yeah, okay. Cytosol and cytoplasm. Okay, all right. Hi. Okay, let's continue then. Anyone who's not able to? So you are supposed to draw this diagram here. Okay. Shall we continue then to ECM? You are supposed to draw it somewhere here. Okay. Shall we continue to ECM? Yeah. Anything you need clarified? Yes. So there's a question. There's a question asking what is the difference between cytoplasm and the cytosol. So actually, cytoplasm and it can be used interchangeably. Cytoplasm actually includes the organelles. Cytoplasm usually includes the organelles, the major organelles. Okay. Cytosol is simply mainly the gel-like material that is found inside the cell. Okay. So that is the simple explanation. Okay. The liquid part of the cell that is found inside is the cytosol. Okay. That you mainly contain water, proteins. Okay. It's the fluid that you have around organelles. So cytoplasm is actually including that as well, including the organelles as well. Okay. So just to get started, okay, shall we move on to extracellular matrix? Okay, then we can do some MCQs afterwards before we move on to cell cycle. Okay, any questions? Okay, extracellular matrix. Come to page number, where are we? Yeah, page number 39. Okay, people, can you hear me? Yes, having dinner. Okay, so I'm not going to have dinner until my this class is over. So you guys can hold on a bit as well. Okay, okay. So okay, so ECM. So this is likely. I mean, so many years it was guessed to be a short note topic. So, but it didn't come. But that doesn't mean we shouldn't practice. Okay, as a short note, this is a very nice topic that you can use to practice. Okay, so I'm going to give it as a homework as well. Okay, so to write a short note on extracellular matrix. Okay, keep that in mind. Extracellular matrix. Let's read. Although animal cells do not have lack cell walls, they have an elaborate extracellular matrix. Okay, so we already know, established that only plant cells, certain prokaryotes and some protists have a cell wall, no? So animal cells definitely don't have a cell wall, but either way, you need something to provide the cell some support, no? The cell should be supported. So in order to do that, animal cells have a very sort of a elaborate and a very complicated external structure outside their cell wall, plasma membrane, that we call as the extracellular matrix. Okay, that we call as extracellular matrix. Main components of the ECM are glycoproteins and other carbohydrate containing molecules secreted by cells. Okay. Then most abundant glycoprotein in the ECM is collagen, which forms strong fibers outside the cell. Now, this is the image I want you to visualize. Okay. So, cytoskeleton, the plasma membrane, as well as your extracellular matrix are all nicely connected so that inside the cell as well as the outside the cell is communicating with each other. Okay. So, now can you see, this is the cytoplasm. So, here you have what? You have your inner supportive structure, those are your cytoskeletal fibers, no? These are all cytoskeletal fibers. Okay, then you have your plasma membrane, which is a phospholipid bilayer. Then you have certain transmembrane proteins. Yes, these are transmembrane proteins. Then can you see outside this plasma membrane, we have this like a network. No, it's like a mesh. It's a network. You can see the dominating thing. Maybe 30 fibers. These sort of big fibers are actually what? Your collagen. These are your collagen. Okay. Collagen is the most abundant glycoprotein in the extracellular matrix. Okay. So, collagen is there. Why do we? So, you know, collagen is a protein. No? We learned that it's a quaternary protein. We learned that collagen is a quaternary protein. So, why are we calling it here a glycoprotein? Why are we calling it here a glycoprotein? Because it is closely associated with carbohydrates. It is closely associated with carbohydrates. So, it is a protein that is connected with carbohydrates. Therefore, it becomes glycated protein. No? A glycated. That means it's associated with sort of carbohydrates. So, you know, we use the term sort of the prefix glyco sort of to describe anything that is associated or related to carbohydrates. No? Okay. So, collagen is the most abundant glycoprotein. What else is there? Collagen fibers are embedded in a network woven out of proteoglycans secreted by cells. Okay. So, this is what we call as, that is the next significant part of the extracellular matrix. You can see here, green color fibers are there. Then out of those green color fibers, you have these sort of branches. This is a zoomed image of that. That is what we call as a single proteoglycan complex. It is composed of a central core of polysaccharide. No, you don't need this, okay? You don't need, you only need to know the name proteoglycan. But just because it's given, I'm just explaining what do you mean by a proteoglycan. So, proteoglycan is like a leaf, no? It's like a leaf that has these smaller, not leaf, ne? It's more like a branch, okay? It's more like a branch that has these sort of leaves that are branching out of it. Okay. So, the proteoglycan has a central polysaccharide, and out of that, to that you have proteoglycan molecules that are connected. So, it has a co-protein as well as a carbohydrate antenna as well. Okay. So, this is what we call as proteoglycan. It's a mix of polysaccharides, proteins and carbohydrates. Okay. So, it's a mix of actually proteins and polysaccharides. Okay. Okay. So, that is the proteoglycan. So, this collagen, we say it is embedded in a network of proteoglycan. It consists of hundreds of proteoglycan molecules attached to a single polysaccharide molecule. Okay. So, what you need to know is the collagen is embedded in a network of proteoglycan. All of this is secreted by the cell itself. So, this is the cell, no? This is the cell. The cell itself secretes them all. Okay. So, this is basically the extracellular matrix. The other thing is, I told you the inner supportive structure, which is your the cytoskeleton, is connected through this transmembrane protein into the extracellular matrix by a special protein. Maybe this is another protein, this is another similar protein. those are called fibronectins. Those are called fibronectins. Okay, collagen fibers are connected to integral proteins. So, this is an integral protein. Integral proteins means what? The proteins that are embedded within the plasma membrane. Can anyone tell me what is the other type of protein that you have in your plasma membrane? Proteins that are not embedded. What do you call them? Proteins that are not within the plasma membrane. What do you call those? Yeah. Yes, methane can you see here? If you zoom in, when a piece of protein is not within the plasma membrane, no, it's not within this bilayer, but it is lightly attached to the inner, or it could be outer. Okay, so that is what we call as a peripheral protein. So anyway, your integral proteins, but mostly it is your transmembrane integral proteins. You know, there's a special type of integral protein called transmembrane protein. So, transmembrane integral proteins connect your cytoskeleton. So, you have the cytoskeleton here. Through those proteins with a special protein, another protein that we call as fibronectin, to the extracellular matrix, so that they are all physically connected. No? So, the external supportive structure as well as the internal supportive structure is physically connected. Okay, so that's going to make the communication between neighboring cells and also to maintain support between neighboring cells all very easy. Okay, because they are all physically connected. Okay, right. So let's write down below this. Here, maybe there's some space here. Let's write down some points. Okay, let's write down some points about the extracellular matrix. Okay, let's write down some points about the extracellular matrix. You can use, so I'm going to use the word ECM, but you can't write that in the paper. Use the full name extracellular matrix. Okay, Hari, even though I use the word short form ECM. Okay, is composed of, extracellular matrix is composed of glycoproteins and other carbohydrate molecules secreted by cells. So, who makes the extracellular matrix? It is this cell. The cell that is surrounded by the extracellular matrix produces the extracellular matrix. Got it? Okay. Okay. Next point. Most abundant glycoprotein is what? These are MCQ points. Is collagen. Next one, this is actually a very important point that you sort of forget after some time. Collagen fibers connected to integral proteins of plasma membrane via fibronectin protein. Then how the fibronectin, who you should now we just learned. What do you mean by fibronectin? This sort of protein pieces that are connecting the plasma membrane proteins to the cytoskeleton and then connecting the cytoskeleton to the extracellular matrix. Those are called as fibronectin. So quickly write this down. Quickly write this down. Okay. Any questions? What's the first point? You can't read. Extracellular matrix is composed of glycoproteins and other carbohydrate molecules. Oh no, right? Secreted by cells. Okay, sorry. Extracellular matrix is composed of glycoproteins and other carbohydrate molecules secreted by the cells. Sorry. Okay. Okay. Shall we do maybe one or two MCQs and then we'll jump to cell division, mitosis. Okay. I'll be sending the question tute that we have done with some of the reviews I have written, because in the up to speed sessions sometimes I might not be able to complete the entire question tute. Therefore, I'll be sending some of the reviews that we have written for the question tute as a PDF, so you can do them by your own and then go through the reviews that we have written to find out the answers. Okay, that is the otherwise. If we don't have time to cover the entire question tute, okay. Where is it? We'll do one or two MCQs from the cellular structures. Then we'll move on to cell cycle. Okay. Can you do this question number 11 and question number 12? Sorry, yeah, question number 11, 12 and 13. All three we'll do. Okay, question number 11, 12 and 13, all three will do. Okay, because these are also mostly from topics that we discussed today. 11, 12, 13. I'll give you 5 minutes, 4 minutes. Okay, quickly do this, Taggala. Okay, and I want you to send me answers as well. Taggala answers. That is the only way I can make sure that you are not sleeping. Okay. Okay, all right. Shall we discuss? Okay, good. I'm receiving some answers. Question two, sorry. Okay, right. It will be delivered to you soon. Okay, so question two. Hold on. Okay, download it from the Zoom chat. Okay, right. What do you think? Question number 11. Which of the following is the best combination of cell junction, its location and function? Okay, so you have to think. Okay, so let's see. If you have a tight junction, that means what is it designed to do? To be sort of leak proof, prevent leakage. Okay, so this is something that was there in your resource book, but after its revision it was removed, so now you have only skin epithelium. Okay, so imagine your digestive system, your intestine. So intestine is a tube like this, and a food particle is moving, and that food particle has been liquefied with all the intestinal juice and the enzymes that you added. Okay, so of course the cells that are in this tube, that is the intestinal lining, should be leak proof. They should not be should be prevented by leakage. Otherwise your abdomen will be full of pieces, full of soup if you had a soup. Okay, or even if something liquefied, not liquefied, but because when you eat maybe rice or something, it will be chewed and then digested and also ground in the stomach, so it will become liquid. Okay, so that will be, your abdomen will be full of liquid if your intestinal cells are leaky, so yes, in your gut, that means your intestine, you have a tight junction. What is their purpose? Is it to communicate? No. So you can put an arrow, you can cut communication, but use a red pen or something, preventing leakage. But if you take anchor junction, so tight junction found in the gut epithelium, correct. Anchor junction, okay, not found in the skin epithelium, also doesn't prevent leakage. Tight junction, gut epithelium prevent leakage? Yes. Most of you are correct. Harry? Very good. Then let's look at the other things. Gap junction, okay, or communicating junctions. Nervous tissue, you can have some gap junctions, okay, but preventing leakage, not the function. Anchor junction, do you have anchor junction, or what is the other name for anchor junction? Desmosomes. Do you have it in the skin epithelium? No. You have in your usually muscles, desmosomes. Okay, so answer number 3. Structure and function, nucleolus ribosome production. What do you think? Nucleolus ribosome production. Okay, not bad. Let's see. Glyoxysomes, photorespiration in plants. That is something we have done in. Now we have covered it in the theory class, but it's given in under the what? Photorespiration in plants is done by a glyoxysome is a special type of peroxisome, I know, but a glyoxysome can't be a peroxisome, no? Sorry, photorespiration is done by a family of peroxisomes, but glyoxysome is a special peroxisome that is designed to do what? Convert fatty acids into sugars. So photorespiration is done by specifically some peroxisome, but you can't take, okay, glyoxysomes are also peroxisomes, you can't take that. Got it? Cytoskeleton determines the shape of the cell. Yes. Provide some support. Vacuoles store soluble substances needed for cellular activities. What do you think? Store soluble substances. Not all vacuoles, not all vacuoles. So because it's not very specific, so if they said central vacuole, yes, you can say store soluble substances, but not any vacuole, because it could be a contractile vacuole, it could be a food vacuole, so not really correct. Produce lysosomes. What do you think? Produce lysosomes? Yes, what do you think? Is it something done by Golgi, producing lysosomes? Yes, producing lysosome. Is it done by Golgi? Yes, that is very correct. So my doubt, so the thing is, few answers are looking like okay, okay. Few answers are looking like okay, but if you stick exactly to the resource, okay. Cytoskeleton, the answer number 3, you can't take exactly, because why? If something determines the shape of the cell, that means something it's fixed, no? So because of the cytoskeleton, you have that shape. Got it? Cytoskeleton doesn't work in that way. Cytoskeleton is a fluid structure. It can break and reform. Okay, it is not something fixed. For example, if you take a cell wall of a plant, it is something fixed. So it is going to maintain a very rigid shape to the, give a very rigid shape to the plant cell, and it won't change. But if you take your cytoskeleton, it is not something very rigid, very firm. It can break and reform. Therefore, you can't say it determines, but it can do what? It can maintain, but not determine. Okay, it can maintain, but not determine. Okay, then nucleolus is actually involving in rRNA production. Ribosomal RNAs are produced in the nucleolus. Okay, ribosomal RNAs are produced in the nucleolus. So that also seems not a bad option. What do you think? So if you have marked, I would take answer number 1 is correct. Second one is not correct, because for respiration you need peroxisomes, not glyoxisomes. Third one, because it says determined, we can't take. Vacuoles, because it doesn't specify it's a central vacuole, you can't take. Golgi complex produce lysosomes is actually the safest answer. Make the safest answer. But if you have written answer number 1 also, I would give marks. Okay. Because the nucleolus synthesizes the rRNA as well as some of the other things needed to make a ribosome. Okay, needed to make a ribosome is actually synthesized in the nucleolus. Where do you have the nucleolus? Where do you have the nucleolus? As the Namimatirane in the nucleus. Okay, so that is the answer for 12. We'll move on to 13. The statements regarding cell junctions are correct. Animal embryos, so this is an ABCD type question, so several answers can be correct, no? Animal embryos have cell junctions which allow the passage of ions. Yes, can anyone tell me what type of a junction? What is the junction that you have here that allow the passage of ions found in animal embryo? Can gap junction, no? Gap junction or communicating junction. What else can pass through this? Ions, sugars, amino acids. Yes. Tight junctions connect the plasma membrane of adjacent cells, forming a continuous seal, preventing leakage. That is very perfect, no? Right? This is the exact same thing that you find in your resource. So that's exactly correct. So A is also correct. Plasmodesmata, I told you, they will ask these keywords. That's what I write them in your in the notes specifically. Okay, the things that you cannot forget, I will definitely give them to you to be written down on the question, the diagram too. Okay, non-living connections between cell walls. No, it's a living connections. No, why is it living connections? You can write here, filled with cytoplasm. Filled with cytoplasm. Therefore, they are living connections. Okay. Desmosomes allow exchange of signals and material between adjacent cells. No. If you have a desmosome, that is designed to give mechanical support by connecting the cytoskeletons. Okay. But if you want to have what? Exchange of signal and material, what do you want? You want a gap junction or a communicating junction. Gap junctions attach cytoskeletons? No. Okay. That is done by what? Desmosomes. Okay. So actually, what are the correct answers? A, B only. Okay, most of you have given good answers. Okay, that's good. Okay. desmosomes. Okay, hurry. Let's go back to the tute. Okay, we are about to move on to the cell cycle. Right. So I'll start with the sort of a software view of what you are going to be seeing and what we are going to step by step go through the so I'm planning to finish mitosis today. Therefore, so let's see, before we actually get into it, what are the, as a quick overview, what we are going to cover stepwise. Okay, so mitosis. Now listen. Okay, now listen up. We are moving on to mitosis. Any question that you want to clarify before that? Yeah, it's a A, B, C, D type question. That means it's the MCQs from 40 to 50. Alright, MCQs from 40 to 50, you have a combination of answers, right? So A and B correct now, is it, I can't remember. If A and B correct now, what is the answer? Then A, C, D nang, there's another answer. B and D nang, there's another answer. That's a combination. So it's given in the paper. Okay. I used to remember in my A-level years because after all the past papers I have done, I could remember the combination even, but it's given in the question paper before the 40 to 50 question set starts. Okay. Right? Okay. Now listen. Mitosis. We are going to move on to cell division. Okay. So, any cell in this world, you learn that, okay, according to cell theory, all cells arise from pre-existing cells. Okay. So, there should be a way the cells that you have right now give rise to new cells. Okay. So, 99.99% of the time in this world, it is an exact photocopying that's going to give rise to new cells. That means, so you have a cell right now. That cell is going to double the DNA it has, and usually double or sometimes even triple or quadruple the organelles, the remaining organelles it has. Then it's going to produce from one cell, you're going to get two cells, daughter. Okay, so we call the first cell that you had, original cell, as the mother cell, and the two other cells, the two new cells, you say as daughters. Okay, so the exact copying or exact doubling of one cell into two, so that it's exactly similar to the mother, is what's going to happen in mitosis. Then in this world, 99 sort of 0.001% of the time, there's a special type of cell division where only happening sexually reproducing organisms, where mother cell is going to produce four cells that are having actually half of the DNA that you had originally. And also those daughter cells are going to be different from the mother as well. That is a special cell division that is called meiosis. That is a bit difficult to or tricky to understand, but once we lay down the foundation in mitosis with the phases and other things, you will be easily able to understand. Okay, so no need to worry. We are going to fill in the tute, then I'm going to stepwise explain one by one. So we'll go through the software and see how this big picture is going to happen. So this is the mother cell. Before everything happens, all the other organelles have been removed for simplicity. This is the nucleus. So keep in mind, in cell division, we are mainly focusing only what's going to happen in the nucleus. Okay. So let's see. So then these are what we call as your centrosomes. Okay. So centrosomes, you can see you had, so this is the original cell with all the organelles. Okay. Now focus on, so all the other organelles are going to disappear. Now focus on the centrosome on the top. It's going to get doubled. So we said originally there's a centrosome located in this sort of region. Before cell division starts, centrosomes are going to get doubled. Then At the same time, can you see what's happening in the nucleus? You have nucleus in the form of uncondensed chromatin fibers in the nucleus, and they are going to get they are condensing on. They are going to condense into a very dense form that we call as the chromosomes. So chromosomes, after they are formed, the covering of the nucleus. What is the covering of the nucleus? What is the outer membrane? Actually, double membrane structure of the nucleus. What do you call that? Nuclear. Nuclear envelope. Nuclear envelope is going to break like this, and then what's going to happen is these centrosomes are going to form like a apparatus. Apparatus means, like, have you seen puppets? Right, puppet shows right, where they are controlled by strings. Puppet shows, or who's the famous puppet? Or Pinocchio, right? It's like that. So these centrosomes are going to produce a sort of a puppeteering wage structure that are going to move all these chromosomes here and there in the cell. That apparatus is called as the spindle. So the spindle, now listen, spindle has, you have your centrosome, which is actually the place where all these fibers, microtubules are arising, and then it has microtubules that are very long, extending forwards, microtubules extending forwards, and at the same time, can you see, there are some microtubules that are extending backwards. Those are called as aster. Short microtubules that are extending backwards are called as aster. This is the main entire spindle. Okay, so we'll see how the entire spindle is formed. Can you see, now this is the spindle is formed so that it's able to control the movement of chromosomes here and there. Then so many things are going to happen. DNA is going to be sort of chromosomes are going to be separated so that it can be given to the two daughters, and so that the DNA is now separated from each other, and then they are going to go into the opposite poles, and then there will be a new daughter cell produced, and the new nucleus is produced, and both of them are going to have separate individual organelles. Okay. Right. Then before we move on to this, I want to give you some basic fundamental understanding about DNA packaging. I think I have taught this. You shouldn't be forgetting at this speed. Before more than the speed of teaching, you keep forgetting. Okay. Now listen. I think I did this just last week. This is the DNA double helix. No? This is how DNA is. This is the currently accepted model of how you have DNA. Then DNA is the molecule, but you have so much of DNA that means inside your nucleus you don't have space to pack all that DNA. What your body is going to do is wrap it around this sort of globular ball like proteins. Can anyone tell me what are these proteins? Around eight such globular ball like proteins. Can anyone tell me what are these proteins? You are going to wrap DNA around it, and then this DNA fragment is going to go and wrap around another set of this protein. I think histone, very good. Right? So this protein complex is composed of 8 molecules of histone protein. So then it's going to package itself like this. Now listen, this arrangement of DNA just wrapped around histone is called as chromatin fiber. Chromatin is a sort of a step 1 of DNA packaging. That means, for example, in your table, if you are studying, that means when you are currently studying, you don't very tightly pack your books into a cupboard, because every day you are studying your resource book. So sometimes you can just stack them on top of each other and just keep them, then you can take them and study. So your chromatin is the first level of packing DNA so that they are easily accessible. So any time you want to see what is in the DNA, you can take them out. But whenever this DNA is about to be divided between two cells, this is not enough, because they have to be very travel friendly. So you have to package your DNA in a very compact manner. That compact manner is going to have several steps of packaging. So imagine your A levels are over, so you are going to give your books to a Malliyo Nangi, okay? So then what you're going to do, you are going to pack them nicely into boxes and then you are going to give them. So this is something like that. When the cell is about to divide, DNA is going to pack into a very small and a very dense structure that we call as a chromosome. Chromosomes are visible under the microscope because they are very dense. Density is high. Okay. So because they are very dense, your chromosomes are visible under the microscope. Okay. So this is a chromosome. If you are a human, how many of these are you going to have? If you are a human, how many of these are you going to have? You are going to have, in your normal cells, you are going to have how many? 46 of these. Okay. 46 of these you are going to have. But now listen very carefully. I said in mitosis you are going to exactly photocopy the DNA that you have right now so that it can be given into two daughters. Okay. So in order to do that, if you have 46 of these, what do you need to do? You need to make copies of everyone. So imagine this is the chromosome number 1. Na? This is chromosome number 1. Na? In order to give to the daughter, two daughter cells, you need to have two copies of chromosome number 1. So these are exactly identical copies. These are exactly identical copies. So they are like twin sisters. They are like twin sisters. Therefore, we call them as kiana. Sister, not chromosomes now, not chromosomes, but it contains two chromosomes. They are now known as chromatids, sister chromatids, because they are like twin sisters. They are identical to each other. Okay, identical to each other, but they are composed of the copy of the same thing. So they are very identical. Okay, so again, if you have 46 chromosomes, you are going to have bigger chromosomes. Let's say this is the chromosome number 2. So you have until about 46 you are going to have, but your sex chromosomes are a bit different. Okay, X and Y chromosomes are a bit weird. Okay, so this could be chromosome set number 2. Then you could have something, maybe a fat and short technique, maybe a fat and short chromosome could be chromosome set number 3. So all these are known as sister chromatids that are connected at their sort of midpoint. That midpoint is known as centromere. So they are connected at their centromere. So that they are like a package. So they are nicely connected with each other until they are separated. Okay. So this is what I want you to understand before we move on to the actual steps of mitosis. Okay, clear. All right, come to the diagram tute as well as open the resource. We are going to go through the eukaryotic cell cycle. Hi, we are going to go through the eukaryotic cell cycle. Okay, come to the diagram tute. Several things we have to discuss. Come to the resource. Eukaryotic cell cycle is divided into two major phases as the interphase and the mitotic phase or the M phase. Highlight interphase is the longer phase, it covers about 90%. Okay, highlight interphase is the longer phase, it covers about 90%. Okay, then interphase could be divided into three phases as the first gap phase or the G1, S phase or the synthetic phase. Synthetic means you are making things. Okay, then G2 phase or the second gap phase. Okay, so we are going to have a nice note here. After explaining everything, I am going to write everything on the board so that you can copy it. Okay, okay. Let's read about G1. G1 could be, I like to remember G1 as the growth phase, first growth phase, but it actually stands for gap. But it reminds you what's going to happen in this G1. It's a growth that's going to happen. Okay, see. In this phase, synthesis of proteins and production of cellular organelles leading to cell growth occurs. Can you highlight? Synthesis of proteins and production of cellular organelles leading to cell growth. Proteins essential for S phase are produced. Then we come to S phase. Very, very important phase. Okay, highlight S phase. DNA replication occurs and synthesis of histone proteins take place. DNA wind around histone beads. So these histones are, because they are like small balls, they look like beads. What do you mean by beads? You know, in necklaces you have on the beads. On the necklaces you have beads. Form chromatin. Okay, so very important phase, S phase. DNA replication occurs and synthesis of histone proteins take place, and DNA wind around the histone to form chromatin. Highlight the entire thing that is happening in the S phase. G2. Cells continue to grow through the protein synthesis as well as cellular organelles. Proteins essential for the mitotic phase will be synthesized. Duplication of centrosomes. I showed you duplication of centrosomes. No? So originally in the cell you have only one centrosome, then it will double. Okay. So we can make a small note here. So you can see M phase. You can see M phase in this diagram. A mitotic phase. In other words, the phase where the actual cell divides into two is a small amount, that is only about 10%. Now, for example, if you take your life, you don't spend most of your life reproducing. Harry, if you are a living, I mean, as a human, you have a childhood, then you have an adulthood, then you have children, then you live your life normally. You don't spend most of it producing children. So a cell is like that. The cell is 90% of the time it is just growing and just being a cell. Okay. So this whole thing, this whole 90% almost is what? Just the cell living normal cell life. Right? So this is 90% is your interface. Then interface has three parts. Interface has three parts. Okay. So I'll just write it here. Okay. Interface. What are things happening in interface? You have a G1. What's going to happen in G1? Protein synthesis. Production of cellular organelles. G1, protein synthesis and production of cellular organelles. S phase, such an important phase, very important. Okay, S. DNA copying, DNA replication. You make photocopies before you produce your two daughters, you need to make copies. You have the entire process of DNA replication under the lesson molecular biology. So don't worry about it right now. You will get enough of DNA copying when you have done molecular biology. Okay. Histone. So I'm actually going to be participating in a special molecular biology camp conducted by our university molecular biology department. So even though I'm doing medicine, I'm going to be participating in a special course. Actually, it's going to occupy most of my Saturday mornings, right? But that's to mainly improve my molecular bio knowledge so that I can teach you guys. Okay, so I'm going to be in the DNA recombination lab and doing PCR and RNA. Okay, got it. So because I feel like I want to improve my molecular knowledge further so that I can teach you guys when you are doing that lesson a bit more better. Okay, so histone production is going to happen in S phase. So they might be thinking, okay, why is there a someone from the medical faculty doing in the applied science department? But I don't care. So I'm just going to go and learn molecular biology, because in medicine you have a sort of a bit of molecular biology, but we have a bit of applied. We only acquire the things that we need to analyze a patient's maybe a report or something, not actual techniques. Okay, formation of chromatin is going to happen in S phase as well. Formation of chromatin. Okay, then I'll write G2 here. Growth continues. in G2. Then most importantly, duplication of centrosomes. Duplication of centrosomes is going to happen in G2. Okay, duplication of centrosomes is going to happen in G2. Right. So don't write yet. I'll give you some more things to be written here. Therefore, you will not be able to manage your space if you start writing right now. Okay. So now you understand. So this interface is just the cell living its life. It's growing, it's copying DNA, it's preparing for its cell division that is coming up. Oh, it's just being a normal cell. Yeah. Like you had a childhood, then you grew up. Okay. So the cell is just living its life. Okay. Let's keep reading, come back to the resource. There are cell cycle controlling checkpoints available at G1, G2 and M phases. Can you see, Metin? There are checkpoints, Metin. Can you see there's a name? It's like a police post. In your school, if at the gates, there are prefects checking your bags, no? There are checkpoints. Can you see G1 there's one, G2 there's one, and at the M there's one. Okay. To ensure that the cell is ready to move for moving into upcoming phases of cell division. So they are making sure that you are mature enough as a cell to divide into two. Okay, you are mature enough as a cell to divide into two, because it's very important that the cell is prepared enough. That means what do you mean by preparation? That it has all the organelles copied, it has the centrosome double. More importantly, we have to make sure that the cell has copied its DNA properly. Properly copying its DNA is so important. If its DNA has not been properly copied, the daughter cells are going to have some problems. So in order to avoid that, there are checkpoints. Okay, there are checkpoints in order to avoid that. Okay. Some cells receive a go-ahead signal at the G1 because that's the first checkpoint. No? Then they will complete G1, S and G2. If it does not receive a go-ahead signal at that point, it may exit the cell cycle, entering into a non-dividing stage called the G note. Okay, so can you see here, if they don't pass, for example, if you didn't get enough results at your first try, what are you going to do? You are going to not be able to go into university. Yes, you are not allowed to, you are not able to go into university that exact time, no? So what do? You attempt it again until that you are stopping your university dream, no? So you are not dream, it's not stopping, but you are not able to continue into university, no? So what do? You again retry. So if you are a cell that is rejected or stopped at the G1 checkpoint, there are two options. Okay, that is only one option is given here. Can you read? It may exceed the cell cycle, entering into an undividing stage called the G node. Most of the cells of the body are in this G node, such as nerve cells and muscle cells. Okay, so if you didn't pass this G1 checkpoint, you enter into G node. Okay, you enter into G node. Actually, your G node can be something temporary even. So if it could be something temporary, so I'll write about G node here, right? So I'll bring this. So you have failed at your G1 checkpoint. So you enter into G node. Okay. So at your G node means you are studying for your second child. That means you are temporarily stopping your journey into a university and you are studying again. So that if a cell is not prepared enough to divide, it can exit the cell cycle and prepare itself again and try to divide again. Okay. So we can write here, if a cell receive a go ahead, can a green light go ahead, traffic light or a green light, go ahead signal at G1, it will enter into G naught phase. Then we want to, something you need to understand is, so sometimes these cells can re-enter, because once they are prepared enough to be divide again, they can re-enter into the cell cycle. But some of the cells given in your in your resource as example, nerve cells and muscle cells, they are something we called as terminally differentiated cells. That means once they divided when you were a baby in your mother's, inside your mother, before you were born, they actually finish dividing and they will never divide again. So they are permanently stuck in the G naught. That's why we say, if a person, for example, a person having a lack of blood to their brain, that person had a stroke. So part of their brain is dead now. So usually those people don't regrow their brain. The brain will not be repaired. That is because brain nerve cells are not able to divide again. Brain nerve cells are not able to divide because they are in a permanent G naught. Okay. So we can write, if the G naught cells are terminally differentiated, that means they are completely, they are specialized, okay, terminally differentiated, they will never re-enter, okay, sorry, they will never re-enter the cell cycle. They will never re-enter the cell cycle and carry out their function until they die. So they will not be able to reproduce. Some people in our society, some people never have children, no? Some of these cells, such as nerve cells and your muscle cells also. If you have a heart attack and some of part of your heart muscle dies, it will never regrow. It will never regrow. Okay, that is because they are what we call as terminally differentiated. Okay, so actually I took some space here also. Here I am going to give you a small overview of the two types of cell divisions, that is actually meiosis and mitosis and meiosis, that you already know, things you already know. Okay, so cell division is of two types. Okay, that is mitosis, that is the one we are learning here, then meiosis. Sorry, these are all level knowledge that I am just reminding. Okay, mitosis happens in what we call as somatic cells, that means your normal body cells, somatic cells. Meiosis happens only in places where reproductive cells are going to be produced. What are your places reproductive cells are produced? If you are a man, in your testes, if you are a woman, in your ovaries. Okay, so in them there are specialized cells designed to produce sperms and eggs. We call them gamete mother cells. So gametes are produced by these gamete mother cells. So here in mitosis, what's going to happen is you have a mother cell and it is going to divide into identical two daughters. Then if you want to talk about the number of DNA or chromosomes, they are going to have the exact number. We call this, they are going to have two copies. That means one copy you got from your father, one copy you got from your mother. That exact same number, we call it diploid. Ne? That means you have two cells, two copies, is going to go into your daughter cells. But if you are a cell that is dividing by meiosis, you are going to have how many daughters? You are going to have 4 daughter cells. But when you are dividing, your DNA amount is going to be half. Because why? These cells are going to become either a sperm or an egg. So if you are a sperm or egg, you are going to combine with another sperm or egg. So if you have full set of DNA, then you are going to have too much DNA in the new organism. No? So when you are producing a sperm or egg, you are going to have half. So this is the difference between mitosis and meiosis. So you can say these are haploid, they can half the amount of DNA. These are diploid. Okay, so I'll give you a few minutes. Can you quickly take everything down in this page number 7 of our diagram tute? Okay. If you want me to zoom in or zoom out, let me know. Okay. Nothing is missing neither. Can you guys see? Let me know. Any question? Today people are there till, so it's 9:30, but people are still in the meeting. Otherwise you guys leave to just to watch recording. So today is good, huh? Okay. So here you write about the interface G1S and G2 here. Then have a summary of two types of cell division, mitosis and meiosis here. Somatic cells do mitosis, gamete mother cells do meiosis. Then I want you to write down a few things about G node, put an arrow to the G1 checkpoint, then bring it here. If a cell does not receive a go ahead signal at G1, it will enter into G node. If the G node cells are terminally differentiated, they will never re-enter the cell cycle and will carry out their function until they die. Examples are nerve cells and muscle cells. If you want me to zoom, let me know. Okay. Any questions, Lama? Under mitosis, it's not clear, understandable. It's the diploid mother. That means the mother is having two copies and the daughters are going to have two copies. Okay, what happened here? Someone drew on my board. Okay, right. Is it okay if I zoom in here? Right, there's an insect walking. Ne? Who's this? Right. He wants to learn up to speed with you guys. Anyway, okay, good. Did you understand about the G note? Okay, G note could be something temporary, G note could be something permanent. So the examples that you have, they have given in your resource for G note cells, such as nerve and muscles, are actually in their permanent G note. They will never re-enter the cell cycle. So I told you, if you are in a temporary G note, if you fail, for example, if you couldn't get into medical faculty in the first attempt, you can go back and prepare yourself and come back. So sometimes some cells are in they enter the G node, then they will re-enter the cell cycle after some time. Okay, so it could be something transient. Again, you have a temporary G node. Okay, temporary G node. Okay, right. Then we can move on to each of the individual phases of the cell cycle. I have actually given here what's actually happening in the G2. That means immediately before you divide. Immediately before the cell enters into the dividing phase, before the reproduction. Okay, so you know, a married, so a person who's married, who's having everything, he's prepared to have a child. Okay, so that is kind of like the G2 phase. He's about to divide by mitosis. So this is what it looks like inside the cell. Metin, can you see the centrosomes have been doubled? Centrosomes have been doubled. So it is all ready to form that apparatus, what we call as the spindle. And then you can see chromosomes have been doubled, because why? You have to give them to two cells. So it is full of chromosome, right? So even though we call it chromosome, the proper way is it's chromatin. No, it is full of chromatin. Okay. Then the nuclear envelope is still there. Nucleolus is still there. Plasma membrane is, I mean, everything is else is normal. Okay. So this is G2 before we enter into the cell division itself. Okay. Right. So come back to the resource. Let's read about mitosis. Okay. Page number 40, mitosis. Okay. Anyone who's still writing this? Anyone who's still writing this? Hands up. Okay, sorry. I'll give you 30 more seconds. Right. Okay. Taggalian. I'm also getting hungry. Okay. If you ever may go into medicine, hunger and your legs hurting, one of the biggest problems you will encounter as a sort of someone in the medical field is actually just your legs hurting, okay, because of all the standing you do, right? All the standing you do. And for me personally, I am standing double, no, triple as one of my colleagues, because I'm also doing teaching. So one of the biggest problems you are going to have with is leg pain. So when we are after ward rounds, there's a doctor's room, okay. So that's actually having this sort of a dirty mattress, and so that we can just go and rest there. Sometimes even my colleagues and sometimes a bit seniors, they just go to the doctor's room, and how they, because of when you stand for a long time, all the blood gets collected in your legs. So that hurts a lot. All the blood gets collected in your legs because you are not sitting down. So what we do is we go to the doctor's room and we just lie down on the bed, and on the wall we have our legs. We are lying down on the bed and our legs are sort of on the wall, so that all the blood can come back towards the rest of the body, because it's very painful when you have a lot of blood collected in your legs, okay, right? So just those are a few things that are happening, I think, okay, because everything is done. You are not doing, you are not going to be doing a sitting down job, such as an engineer or someone who's doing a sitting down job, office job. It's not a work, it's not something you do like that. You have to do ward rounds, you have to walk everywhere in the hospital, okay. That is, if you are becoming a doctor, you don't need to be a doctor. I mean, unless you have the passion to be with people, don't attempt to be a doctor also, because you are going to get tired. Better to go towards a scientist, or even let's say other applied biological sciences, but not towards medicine. If you are not able to, let's say, handle the mental as well as the physical tiredness, okay. But it's satisfaction, it is satisfying, okay. When people get better, it's satisfying, but at the same time it is stressful and it's very tiring, okay. Okay, shall we move on to the resource? Okay, mitosis. There's a definition, page number 40, bottom paragraph. Mitosis is referred to as the nuclear division which gives rise to two genetically identical daughter nuclei from a mother nucleus. That is the definition of mitosis. Please highlight the definition of mitosis. Nuclear division which gives rise to two genetically identical daughter nuclei. There are 5 phases, okay. Prophase, okay. So I'm going to actually, we are going to write under each of these diagrams each of the things that are happening, okay. So first you can see here, it's like a tangled ball of string, no, null ball, tangled ball of string. It is going to be slowly packaged, because you can, whenever you are close to the exam, you have all your books, let's say, thrown around on your desk, no, because you are studying everything, no? So you don't have time to pack it. So that's what it looks like here. But when you are about to give those books to a younger sibling or something, you have to pack them. So that is what's going to happen. So the first thing that is going to happen in prophase, that is the first stage of the mitosis, is you have to say chromatin, that is the uncondensed chromatin, gets condensed to chromosomes. That's the first thing. Second, you can see something different, interesting. No, nucleolus that was involved in making RNA and ribosomal subunits, it's no longer needed, because you need to only divide the DNA. So nucleolus is going to just disappear. Nucleolus disappears. That's the second thing that's going to happen. Third one, I indicated earlier. So once your DNA is doubled, they are going to be connected at sort of their midpoint by special proteins that midpoint is known as the centromere. So remember central, the midpoint is central. Yes, you have to draw this. Yes, you have to be able to draw this. Okay, at the exam, mitosis, the mitosis stages, you have to be able to draw. Okay. Third one. So you know sisters, no? They are identical sisters, no? Twins love, okay? Sister chromatids, once they were copied, when did they get copied? In the S phase. So they got copied in the S phase. Sister chromatids are attached at the centromere. Then you need a whole apparatus. I told you, you need like a puppeteers, controllers, or strings. You need a nice set of controllers so that you can move all these chromosomes here and there. That is called as the spindle. So the fourth thing that is going to happen is formation of mitotic spindle begins. Okay, then I'll because we are not going to be writing anything under this, so I'm just go here. Okay, the mitotic spindle has three parts. The mitotic spindle has three parts. Those are the actual centrosomes. That means, so out of the centrosomes you get the mitotic spindle. So you have centrosomes. Then you have spindle microtubules. They are there strings of the puppeteer wage, hari ar puppet lava control karna strings wage. Then you have our short microtubules that are extending backwards that are called aster. These are aster. These are your proper microtubules. This is the centrosome. Okay. Then final thing that is going to happen in prophase 5, centrosomes start move towards opposite poles because you need one centrosome at one side. Opposite poles. Centrosomes move towards opposite poles. That is the final thing that's going to happen in prophase. Okay. So can you write this down? Don't go into this space actually because we need to write some things here. Can you write under prophase these things? Chromatin gets condensed into chromosomes. You don't need the nucleolus anymore, so it's going to be disappeared. Copied DNA is now present in the form of chromosomes and they are connected at their midpoint, that is called the centromere. Then you need a mechanism to control that DNA chromosome. Therefore, you are going to produce something called the spindle. Then spindle has these three parts. Then the centrosomes are going to move towards opposite poles. Okay. Clear? Okay. Quickly write this. Am I going too fast? Give me some feedback. Is there any homework that we need to check? I forgot no. Is there any homework that we didn't check for a long, I mean, from last week? Okay. Okay. Then we move on to pro metaphase. Okay, but before that, can you jump to the resource? There's some important thing I want you to highlight there. Okay, can you see at the, so we are in page number 41. Can you see? Centrosomes, that final part, centrosomes and centrioles are not available in plant cells. That's so important MCQ point. Okay, centrosomes and centrioles are not available. So therefore, there is no proper centrosomes found in plant cells. Highlight. Spindle is formed during cell division from accumulated microtubule complex. Hurry. So accumulated microtubule complex is a bit of a low version of what is produced by the centrosome. That means, like for example, you can have ChatGPT free version. Okay, ChatGPT free version is there. It is going to do the job of proper ChatGPT, the image generation. Those kind of things can be done by the free version of ChatGPT, but not really well as the paid version. No. So if you take a plant cell, they are going to have something called as accumulated microtubule complex. Okay, highlight that accumulated microtubule complex. So it is going to form the spindle, but it's going to do the job, but not very organized. Like if you take the animal spindle, okay, but in animal cells you have the proper spindle formation by the centrosomes. Okay, so that is something you sort of forget that is given in the resource, could be asked in the MCQs. Okay, then pro-metaphase. Okay, so actually you can understand, so prophase is the one before, then metaphase is the phase after, therefore pro-metaphase is the middleman, no? The phase you have in the middle. So there's, I want you to do some labeling. So there's some interesting things that's going to happen here. Okay, so now listen. First of all, you can see methane. So earlier, what did you have here? Methane. What is this? The nuclear envelope. So you can see here, methane, these are pieces of the nuclear envelope. So the first thing that is going to happen in pro-metaphase, okay, first thing, I'll use blue color, then you can see. First thing that is going to happen in pro-metaphase, nuclear envelope fragments. Okay, nuclear envelope fragments. Second one, okay, chromosomes get even more condensed. Then something very important happens. Okay. Can you see, if you zoom in, maybe theater, okay, you can see it's a bit blurry, okay. You can see in all of these sister chromatids, there's a black color, okay, there's a small part, no? Can you see? That is, so there is a protein, a special protein that anyone tell me what that protein is? What's that protein? So what's going to happen now? Listen, if you have a pair of sister chromatids, a pair of sister chromatids like this, they are connected at their what? The sister chromatids are connected at their what? Centromere. Yes. Then what's going to happen is, these sisters have to be separated. These sisters have to be separated because why? They have to be given to the two daughter cells that are produced. So at a certain point, when these sisters are going to be separated in the middle, from their centromere level, they have to be separated. So in order to separate them, you need to pull them apart. You have to pull them. Okay. So when you have to pull them, you have to hold them somewhere. No? Again, you have to hold them from somewhere. So for that, you have a handle like thing. Remember, one handle per sister, and that handle is connected to a microtubule that is going to be coming from one end and the other one coming from the other end, so that they can be pulled apart. Right? They can be pulled apart. That protein is known as kinetocoprotein. It's a special type of protein. Okay, it's a special type of protein that is going to be attached to the sisters, one for each sister at the centromere. Okay. So that's the protein that you can see here. Okay, so so the sisters have now kinetocoproteins attached to the, so kinetocoprotein is attached to the sisters. Now can you see, so what are these yellow color tubes, okay, what are these yellow color tubes? What are they? These are what? Microtubules, no? Spindle microtubules. Can you see some microtubules are connected to the kinetoco, no? So this one is connected, this one is connected, from this side this one is connected, this one is connected, this one is connected. So can you give me a name to the microtubules that are connected to the kinetoco protein? Something very creative. Microtubules that are connected to the kinetoco protein. Act as an expert biologist. You are discovering that there are kinetoco microtubules. What do you call them? Yes, you are not very creative, no? So you just simply call them kinetoco microtubules. So you can fill in the box here. Okay, microtubules that are connected to the kinetoco are called as kinetoco microtubules. Can you fill in this box? They are kinetoco microtubules. Okay, microtubules. Look very carefully. Can you see? They are connected to the microtubules. Sorry, these microtubules are connected to the kinetoco. Therefore, they are known as kinetoco microtubules. Simple enough. Okay. Okay. Right. Then you can see some microtubules are just not connecting to the kinetoco or to the chromosomes at all. They are just connecting or just going to the other side. No, they are just going to the other side. So as they are not connecting with the kinetoco microtubules, can you give me a name for them? You can simply call them non-kinetoco microtubules. They are simply called as non-kinetoco microtubules. Simple. If you are connecting to the kinetoco, you are a kinetoco microtubule. If you are not connecting to the kinetoco, you are called as a non-kinetoco microtubule. Okay, so we'll write down on the pro-metaphase a few more points. Can you fill in this box, blank box? Third point under pro-metaphase, write down. kinetocoprotein attaches to sister chromatids at centromere. Okay, can you please complete the pro-metaphase set of steps? Okay, nuclear envelope fragments, chromosomes get even more condensed. Third one, kinetocoprotein attaches to sister chromatids at centromere. Okay. You can see, right? Any issues? Prometaphase. Okay. When does the class finish? Okay, you have two options. Do you want me to cover mitosis entirely today, or shall we continue next week? Again, we have metaphase, anaphase, and telophase, and cytokinesis. Shall we finish it next week then? Yeah. What do you do? What do you want me to do? Shall we finish it next week? Then we have tumors and goals to discuss next week, as well as entire meiosis. Entire meiosis is there to be discussed. Okay. What do you think? Yeah. Yeah. Next week. Anything is okay. Okay. Very good attitude. Next week. Okay. Okay. All right. Yeah, I think, I mean, at this point, so I don't want to stress you a lot. So, okay. So I don't want to stress you guys a lot. I'm going to be, I mean, So there are two batches. I am not stressing out right now. That is you guys, because you don't have anything to stress about. And also I'm not stressing out my 26 people. They are, I mean, I finished with them. So I'm also not stressing them out because they have the exam in 10 days. Not 10 days, hold on, 7 days. Okay. So I'm not stressing them or you. But I'm stressing maybe moderately my 27 people because they are entering into their final year. Okay, grade 13. So Hari, okay, we'll continue next week. We have mitosis, but join definitely exactly at 6 next week. Otherwise 6:30 වෙනකන් වල්පල් කතා කරනවා. Then we have half an hour missing. Okay, then we'll finish for the day. Then good night. Thank you for